Very good morning to all. We have completed a vision in special senses. From today, we are going to start ear. Today's class is regarding functional anatomy of ear, external ear, middle ear and inner ear. The SLOs for this topic is to describe the various structural components of human ear, to list the parts of middle ear, to describe the functions of middle ear, to describe the cochlea in the inner ear, to draw and describe the organ of Corti, which is a sense organ. These are the specific learning objectives for today's topic. Coming to the topic. Ear is divided into external ear, middle ear and internal ear. In external ear, it has two parts. One is auricle, also called as a pinna, external auditory meatus. In middle ear, it has auditory ossicles, auditory muscles and Eustachian tube. In internal ear, it has cochlea, compartments of cochlea and organ of Corti. These are the parts of the ear: external ear, middle ear and internal ear. First we will see external ear. External ear consisting of three parts, namely external ear consists of three parts. As I told, it is external, middle and internal ear. This external ear has two parts. One is auricle, that is this cartilaginous part. Then second one is external auditory meatus. External auditory meatus is a tube connecting between the external ear to the middle ear. Now we will see what is auricle or a pinna. It's a part of an external ear which is of fibro cartilaginous plate covered by connective tissue and skin. In many animals, this auricle responds to locate the source of a sound. Coming to external auditory meatus. This meatus starts from the concha and extends inside a slightly curved canal with a length of around 55 mm. This meatus consisting of two parts. One is outer cartilaginous part, second one is inner bony part. What this outer cartilaginous part consists of? This is the initial part of a external ear, external auditory meatus, and is made up of a cartilage. Here is mainly outer cartilaginous is made up of cartilage which is covered by thick skin and contains stiff hair. This hair prevents the entry of foreign particles into the ear. Large sebaceous glands and ceruminous glands also present in the skin, covering this portion of a cartilaginous part of the external auditory meatus. The secretions of these ceruminous glands and sebaceous glands, these secretions along with the desquamated epithelium of a cells which forms a earwax in the ear. That is, a earwax is a secretion of a ceruminous glands, sebaceous glands and desquamated epithelial cell. Next is inner bony part. Inner bony part is a part of the external auditory meatus, is covered by a skin which adheres closely to the periosteum of the bone. The skin covering portion of this is continuous with the cuticular layer of a tympanic membrane. This tympanic membrane is a sign of indication of middle ear. This regarding the external ear. It consists of a auricle or a pinna, external auditory meatus. It has two parts: outer cartilaginous part, inner bony part. Coming to middle ear. Middle ear or a tympanic cavity is a small, irregular, narrow, laterally compressed chamber situated within the temporal bone of the skull. So it is also known as a tympanium. It is separated by external auditory meatus by tympanic membrane. Middle ear has the following structures. One is auditory ossicles, auditory muscles and Eustachian tube. These are the three parts or three materials for a middle ear. Coming to tympanic membrane. This is a diagram showing a tympanic membrane. This is a thin semi-transparent membrane which separates the middle ear from the external auditory meatus. Periphery membrane is fixed to the tympanic sulcus, in which the surrounding bony ring means by fibrocartilage. The periphery is adhered to the tympanic sulcus by fibrocartilaginous structure. This is regarding a tympanic membrane. What is this tympanic membrane made up of? It has a 3 layers. Lateral cutaneous layer, which is a continuation of a skin of a auditory meatus, as I told in the previous slide. It's the external auditory meatus, that is bony part of the auditory meatus which continues with the tympanic membrane. Intermediate fibrous layer which contains collagenous fibers. Medial mucous layers are tympanic mucosa composed of a single layer of a cuboidal epithelial cells. This is a structure of a tympanic membrane. This is regarding a tympanic membrane. Next is auditory ossicles. These are the bones. That is, auditory ossicles are 3 miniature bones which are arranged in a form of chain, extending across the middle ear from the tympanic membrane to oval window. The auditory ossicles are 3. They are malleus, incus and stapes. Now we will see each one. What is malleus? Malleus is otherwise called as a hammer. It has a handle, neck and head. Head is called as a manibrum. It is attached to the tympanic membrane. This is the head. Then neck extends from handle to the head. Then head is capitulum which articulates with the body of the incus. This is malleus which has a handle, neck and a head. Head articulates with the incus. Neck extends from head to the handle. Handle is attached to the manibrum of the tympanic membrane. Coming to incus. Incus is also called as the anvil. The other name for incus is anvil. It looks like a premolar tooth. If you remember premolar tooth, it is similar to the premolar tooth, incus. It has a body, long process and short process. Anterior surface of a body articulates with the head of the malleus, and the long process articulates with the foot plate of a stapes. Coming to stapes. Stapes is also called as a strip up. It is the smallest bone in the body. So it has a head, anterior crest, posterior crest and foot plate. Head articulates with the incus bone and a foot plate fit for a oval window. This is regarding the bones, that is auditory This is regarding the bones, auditory ossicles, that is malleus, incus and a stapes. This is a malleus, incus and a stapes. Coming to auditory muscles. Auditory muscles is of 2 types. One is tensor tympanic, second one is stapedius. What is the function of this tensor tympanic? As the name indicates, tensor is a muscle pulls and keeps the tympanic membrane stretched or in the tensed. It tenses tympanic membrane constantly. So the name is tensor tympanic. It tenses tympanic membrane. This constant stretching of a tympanic membrane is essential for a transmission of a sound waves which may reach a part of tympanic membrane. The paralysis of this muscle, tensor tympani, causes impairment of a hearing. Next is stapedius. Stapedius is the smallest skeletal muscle. There stapes is the smallest bone. Here stapedius is the smallest skeletal muscle in the human body with a length of just over 1 millimeter. It lies in the conical bony cavity and the posterior wall of the tympanic cavity. The function of this stapedius is prevents the excess movement of the stapes. So, when it contracts, it pulls the neck of the stapes backward and reduces the movement of a foot plate against the fluid in the cochlea. So, this prevents the excess of movement of a stapes. So, it reduces the sound waves moving into the middle ear. Then paralysis of this stapedius muscle allows wider range of oscillations of stapes, leading to hyperaction of auditory muscle to sound vibration called as hyperacusis. Even a small sound you are going to hear in the loud sounds when there is a paralysis of stapedius muscle. This is regarding the auditory ossicles and auditory muscle. Coming to tympanic reflex. What is this tympanic reflex? It is also called as the attenuation reflex, characterized by an involuntary contraction of a tensor tympani and stapedius muscle in response to loud noise. Means it reduces the noise from the movement from outside to the inside of a ear. What is the significance of this tympanic reflex? This tympanic reflex, it protects the tympanic membrane from being ruptured from the loud sound, and second, it prevents the fixation of foot plate against the oval window during exposure to the loud noise. Third, it protects the cochlea from damaging effects of loud sounds. This contraction of a tensor tympani and a stapedius during exposure to loud sound develops a stiffness of auditory ossicles so that the transmission of a sound into the cochlea is decreased. This is a protective or a preventive measure for the exposure of a loud sounds to the ear. Coming to third part of a middle ear, that is Eustachian tube. Eustachian tube is also called as a auditory tube. It is a flattened canal extending from anterior wall of a middle ear to the nasopharynx. Its upper part is surrounded by a bony wall and the lower part is surrounded by the fibrocartilaginous plate. So, till now you have read about external ear with a two-part auricle and a pinna, middle ear with auditory ossicles, auditory muscles and Eustachian tube in auditory along with the tympanic reflex. Coming to internal ear. Internal ear or a labyrinth is a membranous structure which is enclosed by a bony labyrinth in pittrous part of the temporal bone. It consists of a sense organs of hearing and equilibrium. Now we will see which are the sense organs for a hearing and equilibrium. In this, the sense organ for hearing is a cochlea and sense organ for a equilibrium is vestibular apparatus. Now we are going to study mainly the cochlear part. This sense organ for equilibrium, that is vestibular apparatus, we will study in the next class, that is physiology of equilibrium. Coming to the cochlea. Cochlea is a coiled structure like snail. It looks like a snail shell. It consisting of two structures, that is central conical axis formed by a spongy bone. Central conical structure which forms a spongy bone called as a modulus. Bony canal or a tube which winds around the modulus. That is bony canal or a tube which winds around this modulus. Central conical axis with a canal which winds around the modulus is cochlea. Now we will see the compartments of a cochlea. Two membrane partition extend between the osseous spiral lamina and the outer wall of the spiral canal. Both the membranes divide the spiral canal of the cochlea into three compartments. The membranes of the cochlea are basilar membrane. This is a basilar membrane, and second one is vestibular membrane. This is a vestibular membrane. This is a diagram showing a cross section of a cochlea where it has a two membranes which divides a cochlea into three compartments. The two membranes are basilar membrane, this brown color one, and this is a vestibular membrane. Now we will see what is basilar membrane. This basilar membrane is a connective tissue membrane. It stretches from the tip of the osseous spiral lamina to tough, dense, fibrous end of the spiral ligament. Spiral ligament, from the tip of the spiral lamina to the tough, dense, fibrous band called spiral ligament. It stretches from either end to divide the compartment into two parts. Coming to vestibular membrane. This vestibular membrane is also called as a regional membrane. You should remember the other names because in exam they may ask you either names for a 3 marks or a 5 mark question. So it is a thin membrane. It is placed obliquely between upper surface of the spiral lamina and upper part of the spiral ligament. Compartments of the, so these are two membranes which divides a cochlea into three compartments: basilar membrane and vestibular membrane. So which are the three compartments divided by these two membranes? One is scala vestibuli, second scala media, this one is scala tympani. The compartment, first one is vestibular, before the vestibular membrane it is scala vestibuli. In between the vestibular membrane and basilar membrane it is scala media. Here it is scala tympani. Now what is this all three compartments filled with? All three compartments are filled with a fluid. In order to transmit a sound waves from air to water media, so this middle layer consists of a fluid, that is scala media, scala vestibuli and scala tympani. Both are filled with a perilymph. Scala media is filled with a endolymph. So scala vestibuli and scala tympani is filled with a perilymph, whereas scala media is filled with a endolymph. This is a same diagram showing in this way. This is scala vestibuli continuous with the scala tympani. The middle one is the scala media. As I told in the first slide, the foot plate of the stapes, these are the auditory ossicles, malleus, incus and stapes. This foot plate of the stapes covers over the oval window of the scala vestibuli. The fluid is perilymph in the scala vestibuli and scala tympani, and scala media it is endolymph. Coming to scala vestibuli. This scala vestibuli lies above the scala media. It arises from oval window which is closed by the spiral canal of the cochlea of the foot plate. This diagram, this scala vestibuli which has a oval window which is closed by the foot plate of the stapes. It is closed by the foot plate of the stapes. So it allows the bony canal up to its apex. At the apex, it communicates with the Celiac tympani through a small canal called helicotrema. At the end, it communicates with a small gap with the Celiac tympani, which is called as a helicotrema. What is this? At the apex, it communicates with the Celiac tympani through a small canal called helicotrema. So this is regarding a scala vestibular. Coming to scala tympani. Scala tympani, which lies below the scala media. It is parallel to the scala vestibuli, ends at the round window. This round window is closed by the strong thin membrane known as a secondary tympanic membrane. This diagram shows that this scala vestibuli continues with the scala tympani, which has a round window which is closed by a thin membrane called secondary tympanic membrane. It has a fluid that is perilymph. I repeat, scala vestibuli continues with the, at the apex it has a small opening to connect to the scala tympani. The opening is called helicotrema. The scala vestibuli has a oval window which is covered by the foot plate of the stapes, continues with the scala tympani, which has a round window covered with a thin membrane, that is secondary tympanic membrane. That is regarding the scala tympani. Coming to scala media. Scala media is otherwise called as a cochlear duct or membranous cochlea or otic cochlea. It has a triangular compartment enclosed by the basilar membrane and vestibular membrane. This is a triangular compartment enclosed by the basilar membrane and the vestibular membrane. The upper surface of the basilar membrane is epithelial cells arranged in the form of special structure called organ of Corti. It is a sensory part of the cochlea. It is called as a otic. This scala media is called as a otic cochlea or cochlear duct or the membrane as cochlea because it has a special structure called organ of Corti, which is a sense organ for a hearing, which lies above the basilar membrane. Coming to organ of Corti, which is a specialized structure which is called as a receptor organ of a hearing. It is a neuroepithelial structure in the cochlea because it has a epithelial cells and it continues as a cochlear nerve. This is a neuron part and epithelial cells which form this structure. This is organ of Corti. We will learn in detail regarding this organ of Corti. This organ of Corti, where it is situated? It is situated above the basilar membrane. As I told in the previous diagram, it is above the basilar membrane and it extends from lip of the osseous spiral ligament and the basilar membrane. It extends to the cochlear duct. Except for a short distance on either end, it moves throughout the cochlear duct. Roof of the organ of Corti is formed by the gelatinous or a tectorial membrane. This roof is formed by the tectorial membrane, which is made up of a gelatinous material. Coming to structure. Organ of Corti is made up of a sensory elements called hair cells and various supporting cells. In vision, we have a receptors, rods and cones. Likewise, in hearing, we have a receptor organs, hair cells and various supporting cells in this organ of Corti, which helps for a hearing. All the cells in the organ of Corti are arranged in the from center to periphery of the cochlea. It is from the center to periphery of the cochlea. The cells are arranged in this form. There are around 10 cells from center to the periphery. First one is a border cell. This is assume this is the center and this is the periphery. From here we will start the 10 layers of organ of 10 cells of organ of Corti. First is border cells. This one is a border cells. Second is inner hair cell. This white color flash shaped inner hair cells of 2 rows. Then inner phalangeal cells. These are all the inner phalangeal cells. Then inner pillar cells, outer pillar cells, outer phalangeal cells, outer air cells, cells of Hensen, cells of Claudius and tectorial membrane and the reticularis. Now we will see in detail what are these structures. I repeat from center to the lateral. You should remember this. This may be asked for a part of a main question or a 5 marks. It is a very important structure. That is, first is a border cell. This small only one is a border cell. Second, inner hair cell. This white flash shape. Then inner phalangeal cells, inner pillar cells, which is attached to this inner phalangeal cells, outer inner this in between this inner phalangeal and outer pillar cell, it is a tunnel. Inner tunnel is here, which you learn in detail in the next slides. Then these are the outer phalangeal cells, outer air cells. This is the outer tunnel in between the outer air cell and the cells of Hensen, then cells of Claudius. The top layer of organ of Corti is the tectorial membrane and lamina reticularis, which is formed with a gelatinous material. Coming to each one. Border cells. This border cell are slender columnar cells arranged in a single layer on the tympanic clip along with the inner side of the air cell, inner air cells. Okay, the surface of the borders have a cuticle. Okay, this border cell is a slender columnar cells arranged in the single layer and tympanic membrane along the inner side of the inner hair cell is a border cell. Coming to inner hair cell. Inner hair cells are flash shaped cells. There are 2 here. They are flash shaped cells and are broader than the outer hair cell. Compare here. This is the inner hair cell. This is the outer hair cell. These cells are flash shaped with a broader than the outer hair cell. These inner hair cells are arranged in a single row, occupy the upper part of the epithelial cell layer. They have a round base. Each cell rests on adjacent supporting cells called inner phalangeal cells. It has a round base which rests over the inner phalangeal cells. But they are flash shaped. They are broader than the outer air cell and they are arranged in a single row, occupy the upper part of the epithelial cells. They have a round base which rest over the supporting cells for the inner pharyngeal cells. This is regarding the inner hair cells. These inner hair cells, they have a number of short stiff hair which are called as a stereocilia. Each hair cell is around 100 stereocilia. See, this is a complete hair cell. It has a number of stiff hairs at the top. They are called as stereocilia. Each hair cell has 100 stereocilia. One stereocilia which is larger is known as a kinocilium. See the arrangement here. The longer one is a, larger one is a kinocilium. Stereocilia are in contact with the tectorial membrane. Remember this, here you can't see in that much clear. So this is a part which we have taken there. So this stereocilia, they are in a constant contact with the tectorial membrane. These stereocilia and kinocilium are in contact with tectorial membrane. Inner head cell and the outer hair cell together form the receptor cells. The sensory nerve fibers are distributed around this hair cell. Both inner and outer hair cells have a afferent nerve ending and efferent nerve ending. Okay. This each hair cell has 100 stereocilia. They constantly contact with the tectorial membrane. Each has a sensory nerve fibers of afferent and efferent nerve fibers. This is regarding the inner hair cells. Coming to inner pharyngeal cells. These are inner pharyngeal cells are the supporting cells of inner hair cells, because inner hair cells, they rest over the inner pharyngeal cells, so they form the supporting cells for the inner hair cells. They are arranged in along the inner surface of the inner pillar cells. See here, this is the inner pillar cell. They are arranged like a row inside inner surface of the inner pillar cells. Their bases rest over the vascular membrane of the spiral ligament. The inner pharyngeal cells base rest over the basilar membrane. The inner hair cells rest over the inner pharyngeal cells. So this inner pharyngeal cells forms the supporting cells. Coming to inner or outer pillar cells. They are called as rods of Corti. See, this one is the inner pillar cell, outer pillar cell. Each pillar cell has a broader base and an elongated body or pillar head at the tip of the pillar. Bases of the pillar are closed with a spiral lamina. They are broader base with elongated pillar or head tip of the pillar. Inner pillar cell, this outer pillar cell, their bases are broad. They rest over the spiral lamina. These are the inner and the outer pillar cells. These pillar cells, inner and outer pillar cells, slope towards each other. Their heads articulate. Thus, the pillars of the cell form a series of arches which enclose the triangular tunnel or an inner tunnel called as tunnel of Corti. This inner pillar cell, outer pillar cell, which cells slope towards each other and their heads articulate, which forms a series of arches which enclose a triangular tunnel which is called as a tunnel of Corti. This triangular area which is called as a tunnel of Corti or inner tunnel. Coming to outer pharyngeal cells. These are all the outer pharyngeal cells. Outer pharyngeal cells is also called as a Ditter's cells. D-E-I-T-E-R-S, that is Ditter's cells, which are the supporting cells for a outer hair cell. See, inner pharyngeal cells are the supporting cells for the inner hair cell. Outer pharyngeal cells are supporting cells for the outer hair cell. Outer pharyngeal cells are tall columnar cells. Coming to outer hair cell. What is this outer hair cell? These outer hair cells are the columnar cells occupying the superficial part of the epithelium of organ of Corti. Their bases are supported by, or they have a support of outer pharyngeal cells. The rest over the outer pharyngeal cells. The structure of outer hair cell is similar to that of the inner hair cell, except they are not broader as that of the inner hair cell. Okay, that is regarding the outer hair cells. Coming to cells of Hensen. These are all the cells of Hensen. They are the tall columnar cells forming the outer border of the organ of Corti. They form the outer border. This is the center, this is the periphery. So this form the outer border of the organ of Corti. These cells are arranged in the several rows on basilar membrane lateral to the outer pharyngeal cells. They are tall columnar cells. They arranged in several rows. They are not in a single row. They are in the several row on the basilar membrane lateral to the outer pharyngeal cells. The space between the outer pharyngeal cells and the outer hair cell are outer tunnel. This one is outer inner pillar and outer pillar cell. The space is inner tunnel. Here cells of Hensen and outer hair cell, the space is outer tunnel. The space between this is outer tunnel. This is regarding the cells of Hensen. Coming to cells of Claudius. These are all the base ones are the cell of Claudius. They are the cuboidal cells. These are all the tall columnar cells. Cells of Claudius are the cuboidal cells in nature. They line the lower surface of the external spiral circus. In certain areas, they form the group of cells present between the cells of Claudius and the basilar membrane. They are the cuboidal cells which rest over the basilar membrane. This is the cells of Claudius. Coming to last one, that is tectorial membrane and the lamella reticularis. It extends from the vestibular lip to the level of cells of Hensen. Extends from level of cells, say from vestibular lip to the level of cells of Hensen. It extends from there. It forms the roof of organ of Corti. So cuticular plates of supporting cells. They are phalangeal cells, pillar cells, Hensen cells and Claudius cells are the supporting cells which collectively form the reticular form known as a lamina reticularis, which covers the whole organ of Corti. This is regarding the organ of Corti. I will just repeat superficially regarding this layer of organ of Corti. Cells arranged from center to periphery. First is border cells, second is inner hair cell which rest over the inner phalangeal cells, then coming to inner pillar cells, outer pillar cells. The space between this inner and outer pillar is outer tunnel or tunnel of Corti, then outer pharyngeal cells, outer hair cell, cells of Hensen, cells of Claudius. All this rest over the basilar membrane and the roof of this organ of Corti forms a tectorial membrane. Okay, this regarding the organ of Corti, which is a sense organ of a hearing. Coming to impedance matching. What is impedance matching? As you know, the sound is moving from the air to the fluid medium. The air filled in the middle ear conducts sound waves mechanically into the fluid filled internal ear through the ossicular system. The effective transfer of sound energy from an air to fluid medium is difficult because most of the sound is reflected from the surface of the fluid medium, the outside, so because of which impedance mismatching will occur. This fact can be studied by observation that when a person is below the water, when you speak outside, so it won't be heard because this happens because 99% of the sound is reflected from the surface of the water because the impedance offered by the water. Exactly similar situation, that is air from the air filled middle ear transmits the sound to the inner ear which is filled with the endolymph and the perilymph. The impedance offered by ear is endolymph and the perilymph for a transmission of a sound from the middle ear to the inner ear. So to overcome this, the impedance matching mechanism is established by the middle ear. So what is this impedance matching? The middle ear functions impedance matching device primarily amplifies the sound pressure. It is accomplished by the three mechanisms. So this diagram already you know, this is a tympanic membrane, malleus, incus and stapes. So these are all the ones which help for a impedance matching. The three mechanisms which maintains the impedance are lever action of ossicles, that is malleus, incus and stapes, which has a handle of malleus is 1.3 times longer than the long process of the incus. This provides a mechanical leverage advantage, that is due to which a middle ear ossicles increases the force of movement by 1.3 times. I repeat, it is a handle of malleus is 1.3 times longer than the long process of the incus, which increases the middle ear force of movement 1.3 times. It is a lever action of the ossicles. Coming to hydraulic action of the tympanic membrane, which is exerted because effective vibratory area of tympanic membrane is around 45 mm square. The effective vibratory area, the handle of the malleus is attached to the center of the tympanic membrane. So the effect of vibratory area of tympanic membrane is around 45 mm square, is much greater than the stapes which covers the oval window, that is around 3.2 mm square. If you remember, the foot plate of the stapes which covers the scala vestibuli in oval window. This oval window is a smaller area which is covered by the foot plate of the stapes. That is, the area of tympanic membrane vibratory area is around 45 mm square, and the oval window which is covered by the foot plate of a stapes is around 3.2 mm square. This size difference, force produced by the sound is concentrated over the smaller area. The vibration is produced for the larger area and it is concentrated to the smaller area. Thus the amplifying the pressure exerted on the oval window is around 18 folds. Okay, it is around 18 folds the pressure exerted near the oval window. This is a hydraulic action of a tympanic membrane. Coming to curved effect of a curved membrane effect. The movement of the tympanic membrane are more at the periphery than the center. That is the center where the handle of the malleus is attached. This is to provide some leverage action for a impedance matching. The periphery of the tympanic membrane vibrates more than the center of the tympanic membrane because of attachment of the handle of the malleus. So we will see how these values will come. So effective vibratory area of the tympanic membrane is around 45 millimeter square. The foot plate area which covers the scala vestibula is 3.2 millimeter square. So the area ratio is 14:1 ratio for tympanic membrane and the foot plate of the stapes. The lever ratio is ossicle ratio, the handle of the malleus to the long process of the incus. It is around 1.3:1 ratio. So the total transformer ratio is 14:1.3, that gives around 18:1 ratio. The pressure at the foot plate of the stapes, that is covered by the oval window, is covered by the foot plate of a stapes. The pressure exerted here is around 18 fold. This is how we got a values. I repeat, this is the effective vibratory area of the tympanic membrane is around 45 millimeter square. The foot plate area is around 3.2 millimeter square. So the ratio between the tympanic membrane and the foot plate of a stapes is 14:1. The lever ratio, that is the handle of the malleus, is 1.3 times longer than the long process of the incus, that is 1.3:1. So the total transformer ratio is 14:1.3 is around 18 times the pressure near the oval window near scala vestibula, which is covered by the foot plate of the stapes. So this is the topic for today. Any doubts you can post.
morning students. You're welcome to the next very important class of molecular biology where we'll be discussing regarding the regulation of gene expression. You all very well know that DNA is the chemical basis of heredity and it is the reserve bank of genetic information, okay, and it is composed of various functional units called genes. We are stressing this because there are also certain units which are not functional or whose function is not known. Those are known as orphan genes, okay. Right now we are interested only in functional genes which have some purpose to serve. Now what do you mean by genome? It is the total genetic information contained in a cell, okay. So we say that the genome is constant in that human being. What we mean by this? So if genome is the total genetic information contained in a cell, so irrespective of the type of the cell, whether it's a hepatic cell or a cardiac cell or some other cell, brain cell, etc., it has to contain the same genetic information. And yes, it is true, all the cells in a particular human being or a particular animal contain the same set of RNA, the same set of chromosomes, and the total genetic information is very much same. But how is it that liver performs different function, the brain performs different function, muscles differently? It is because not all the genetic information stored in this genome is expressed uniformly in all the cells, okay. Depending upon the specific functions of each of the organ or tissue, only those genes are respectively expressed in such tissues, okay. So here assumes the importance of regulation of gene expression. Before going on to know what regulation of gene expression is, you should first know what gene expression is. We all know the central dogma of life, wherein DNA undergoes replication to form the daughter DNA molecules. DNA in the next process will form different types of RNA molecules, okay. It doesn't stop here because the end product which is utilized by the tissues in the body is neither DNA nor RNA, but it is proteins, right. So these RNAs have to be translated into proteins for them to be the functional forms, okay. This is nothing but gene expression. What part of gene is being expressed or what part of gene is being functionally available or is being translated into protein is nothing but gene expression, okay. Like how you studied the regulation of various metabolic pathways in carbohydrate, lipid, protein and nucleotide metabolism, you also have regulation of the gene expression per se. As I told you earlier, all the genes cannot or otherwise need not express uniformly in all the tissues, okay. So depending on the requirement of the body and depending on what that particular organ or tissue is designed for, okay, the respective genes are expressed, okay. And in order to check that these are not produced or these are not active throughout the day, okay, there is something known as regulation, because all these processes are highly energy consuming. Both transcription and translation are very energy demanding processes. So we do not want to unnecessarily spend energy producing all the proteins from all the genes possible every time, right. It is only selectively produced wherever and whenever it is required. So to make sure that it is contained, regulation is very important, okay. I hope you understand. So gene expression refers to synthesis of proteins under the influence of gene. So it is a combined process of transcription and translation. So not everything that is coded in DNA is expressed, and even if it is expressed, it is not uniformly so in all the tissues or in all the cells of the body, okay. I think I am clear with this in the fundamental concept. Now you already studied about mutation. What is mutation? It is change in the nucleotide sequence of DNA which alters the regulation or expression of a gene, which finally results in the synthesis of a dysfunctional or non-functional protein. It can be totally non-functional, the protein which is produced, or it can function in a different way which is not required in that particular instance. So it is dysfunctional or non-functional protein. Thus, the product of gene expression is finally protein. It can be enzymatic or non-enzymatic. Okay. So here you can note that there is a DNA molecule here which is transcribed to form different types of RNA. Following post-transcriptional modifications, you have the mature mRNA which is still in the nucleus. Okay. It is only during the process of translation that this mRNA is thrown out of the nucleus, where it comes and sits on the rough endoplasmic reticulum. With the help of rRNA and tRNA, it starts synthesizing the protein via a process known as translation. And as you know, this protein is a native protein which is synthesized following translation. It is not used up directly in the tissue of interest, rather they are modified according to needs of the body. So the set of processes which convert this native protein into the usable form of protein are known as post-translational modification. Okay. So as I explained you, first you should know what is gene expression, and then what do you mean by regulation of gene expression and why is it needed. Okay. So the rate of expression of prokaryotic genes. In prokaryotes, the genes are mainly controlled at the level of transcription, that is the formation of different types of RNA from DNA. At that point itself it is regulated. But however, in eukaryotes, the genome is much larger and much complex. So you cannot expect the similar simple way of regulation in eukaryotic genes. So it is regulated by many ways. So since the genome is larger and complex, even the regulation of genes is complex in eukaryotes. Okay. Now, why is it required at all? Why should we regulate gene expression? Because it is required for growth, development, differentiation, metabolic functions and existence of an organism, and most important is to avoid unnecessary spending of energy and resources synthesizing all the proteins in all the tissues all the time. Now, how are genes regulated? Once you know that the genes are regulated, you also should know how are they regulated. As you know, with respect to other regulations, even here it is positive and negative regulation. Positive regulation leads to stimulation of the genes to express, okay, so that the protein is formed, okay, whereas negative regulation is suppressing the genes to make sure that unnecessary proteins are not synthesized. Okay. In positive regulation, the expression is increased by a regulatory element, okay, known as inducer, also known as the positive regulator. Okay. In case of negative regulation, when you don't want that gene to be expressed, okay, certain molecules known as negative regulators or repressors do come into action, okay, and suppress the expression of gene. Okay. So broadly, the genes can be regulated positively when they are needed, negatively when there is no need. So the types of genes, not all genes are similar, because there are certain genes which are expressed only when an inducer is present. They want a stimulus for it to express. I think this can be compared with most students in the class, where unless we ask you to study, unless we conduct internals, unless we give you assignments, you do not study. You take it for granted that listening to the class itself is enough. Okay? Okay, right? So these are inducible genes like most of the students. Okay? For example, the beta galactosidase enzyme is induced by lactose in prokaryotes. This enzyme, as it says, it is an enzyme which will cleave lactose into galactose and glucose, helping in utilization of lactose. Okay. So this is an inducible gene, and insulin induces the gene for glucokinase in human beings. So this is not all the time induced in human beings. Okay, only when it is required, the insulin will induce the gene for this glucokinase, and only then it is produced. These are the set of genes known as inducible genes or which need a stimulus for them to express themselves. There are other sets of genes like a minority in our class, okay, which constitutively express, that is, this expression of these genes is not regulated. They don't need any stimulus to express themselves, okay. Rather, they are expressed at a constant rate. There are few students who regularly study, right, and update their assignments. Likewise, okay, you have constitutive genes. For example, most of the glycolytic enzymes are constitutive in nature, okay. They are often referred to as housekeeping genes because they are very much required for day-to-day activities of life. So, they are not regulated, rather they are constantly produced. Now, how is it that the regulation of gene expression is happening in prokaryotes? We have, as I told you, the regulation is different in prokaryotes and eukaryotes. Let us learn the simpler thing, that is the regulation of gene expression in prokaryotes first, then we will move on to the regulation in eukaryotes. A concept known as operon concept has been proposed in order to explain the regulation of gene expression in prokaryotes. Okay. Now, what is this operon? In prokaryotes like bacteria, the genes involved in a metabolic pathway are often present in a linear fashion, which is very simpler, and all these sets of genes which are involved in regulating the expression of prokaryotic genes is known as operon. Okay, it is not a single entity. It is a cluster of genes, okay, which helps in regulating the expression of genes in prokaryotes. Okay, so to summarize, operon can be well defined as a coordinated unit of genetic expression in bacteria. Okay, so when will coordination happen? When there are multiple subunits. So basically it contains multiple genes and they do not act independently but rather in a coordinated fashion. So all these genes act in a coordinated fashion and it is collectively known as operon. Okay. This concept was introduced by Jacob and Monod when they were studying the regulation of lactose metabolism in E. coli. Okay. So how this is the basic mechanism to help us study the regulation of gene expression in prokaryotes. Now, what is this lac operon? Lac stands for lactose. Operon, as you know, is a coordinated unit of genetic expression, right? A set of genes which are required for metabolizing lactose, okay, are situated in a region and they are acting in a coordinated fashion, okay, to regulate the metabolism of lactose, okay, by processes known as repression and derepression, okay. This entire concept is known as lac operon concept. We will go into the details of it. This lac operon concept is a model for studying the regulation of lactose metabolism in E. coli. Okay, so it is a coordinated unit of gene expression to make or synthesize the enzymes which are necessary for lactose metabolism. I told you that we need beta galactosidase in order to split lactose into glucose and galactose, and then utilizing it when the energy needs demand. Okay. So in order to make those enzymes which help in metabolizing lactose, okay, there is a coordinated unit of genetic expression known as operon, and this is specifically known as lac operon. Okay, this is the structure of lac operon, where you can see I, P, O, the three genes, that is I for regulatory gene, P for promoter site, and O for operator gene. These three are the regulatory genes. You also see that there are structural genes Z, Y and A. These structural genes are individually involved in producing each of the enzymes required for lactose metabolism. In turn, these structural genes are regulated by the regulatory genes I, P and O. We will go into a little detail. Okay. So, the regulatory gene, which is represented by I, produces a repressor protein. Again, how? Through the process of transcription and translation. Okay. This part of the gene alone will produce a specific mRNA, which can translate into repressor subunits. Repressors are basically negative regulators, right? They do not let you express the gene, right? It is a tetrameric protein, repressor protein, okay? What it normally does is, it goes and binds to the operator gene, okay? I told you that these three are regulatory genes, and promoter site, okay, is the site which helps the RNA polymerase, okay, to find the site where the transcription has to start, right? The initiation of transcription will be identified by the RNA polymerase with the help of this promoter site. Because this promoter site is sitting adjacent to this operator gene, it gets influenced by what is happening in the operator gene, okay? So, when the repressor proteins produced by the regulatory gene will go and sit in the operator gene, it negatively influences the promoter site, so that it withholds from helping the RNA polymerase to for it to find the transcription initiation site. Only when it starts transcription it can produce a RNA and then protein, right? So it arrests the process at the start of the transcription itself, okay? So this P site helps helps the RNA polymerase to find the transcription initiation site, okay, which is normally not happening because of the presence of repressor protein in the O region, and this being produced by the I region itself, okay. So this is the coordinated activity which is not letting the structural genes produce the necessary enzymes. Now what is the operator site doing? The regulatory protein which is produced by the regulatory gene, okay, is will bind to this operator site and blocks the initiation of transcription. Hope you understood. Is there any doubt? If yes, please ask, I will explain again. Okay, if there are no doubts, I will continue. Okay. So this is another representation of the same lac operon concept, wherein you have the regulatory genes I, P and O, and you also have the structural genes Z, Y and A, okay. The structural genes can produce the respective enzymes of lactose metabolism only when it gets a signal from the regulatory genes, right? The structural gene Z codes for beta galactosidase, Y codes for permease and A codes for transacetylase, okay. Each of these are required for metabolizing the lactose, okay, in E. coli. Now all these three structural genes transcribe into a single large mRNA. You can see that the final enzymes are three different, but the mRNA which is producing these three different enzymes is single, right? So this mRNA is produced as a single large subunit or single large mRNA by the three different structural genes, okay, and this is known as polycistronic mRNA. This is very rich in prokaryotes. What do you mean by polycistronic? Each of the part of mRNA will code for different protein or different enzyme, okay. The entire mRNA which codes for one single protein is known as cistron, okay. So here we have three different cistrons in the same mRNA, right? It is known as polycistronic mRNA. It is the single mRNA which codes for three different enzymes, okay. And prokaryotes are very much rich in polycistronic mRNAs. Now what do you mean by repression of lac operon? As you know, repression is nothing but the negative regulation, right? It is a mechanism by which the presence of excess product of a pathway shuts off the synthesis of its key enzymes. It is very much comparable to the regulation in other metabolic pathways, where there is product inhibition. When there is too much of product in a metabolic pathway, it goes and inhibits the enzymes prior to that product, so that there is no continuation of the pathway, right? It is the similar fashion which is applicable to lac operon concept also. Okay, as I explained, this is a constitutive gene which keeps on producing a specific mRNA, which translates into a repressor protein, which goes and binds to the operator site. Okay, so that the promoter site will get influenced by the presence of repressor protein on the operator site, and it doesn't perform its function. What is the function of promoter region? It is to help the RNA polymerase to find the transcription initiation site. So normally it doesn't happen. Okay. So this is what happens in the absence of E. coli or in a normal condition in E. coli. Okay. But what happens in presence of lactose? Suppose there is lactose in the medium, or the person or the bacteria has consumed lactose. Okay, it enters the E. coli. Okay, so the repressor molecules which was binding O has high affinity for lactose. Okay, so outside this entire structure is too much of lactose available. Okay, in presence of excess lactose, this lac repressor protein has high affinity for lactose. Okay, so that it goes and binds to lactose. Lactose molecules bind to this protein and induce a conformational change in the repressor protein itself. Okay, so that the repressor gets inactivated and it withers off from the operator gene site. Okay, it is no more blocking operator site, because now it developed too much of affinity for lactose. It's just a competition of affinity between the lactose molecules and the operator site. Okay, in the absence of lactose, it has no other option. It had to go and sit in the operator site. But when lactose is given, it prefers to go and bind with lactose. Okay, so it moves away from its site. Okay, it goes and binds to the lactose. So the inhibition laid on the P site is relieved. Okay, this is known as de-repression. Okay, repression when there is no lactose, de-repression or release of inhibition when there is lactose. Okay, so in that case, when there is release of inhibition at the O site, P site will get positively influenced. Okay, and then it helps the RNA polymerase to attach to the P site. Okay, and then the transcription proceeds, leading to the formation of polycistronic mRNA. That is, only when this O site is free and the P site is positively regulated, it helps the specific RNA polymerase to read these structural genes Z, Y and A and transcribe it into a mRNA. Okay, polycistronic mRNA. Okay, when that mRNA is synthesized, okay, you also have the production of the specific enzymes beta galactosidase, permease and transacetylase. So here the lactose acts by inactivating the repressor molecule. The repressor molecule had a negative influence. You are inactivating the negative influence. So negative into negative becomes positive, right? So by removing the inhibition, it is acting as a inducer or it is acting as a positive regulator. So this entire concept is known as de-repression of lac operon. Repression in absence of lactose, de-repression when there is lactose. So after this, we need to know that E. coli cells, like many other cells and tissues, they prefer utilizing glucose as a fuel with reference to, in preference to lactose. Okay, so when you give both these fuels to E. coli, it prefers using glucose. Okay, it is only after the glucose is depleted that the utilization of lactose starts from E. coli. This somehow indicates that glucose can interfere with the induction of lac operon. when there is induction of lac operon concept in presence of too much of lactose, there is stimulation of synthesis of enzymes required for lactose metabolism. Okay, so when it is not happening in presence of glucose, okay, or in presence of both, okay, it gives a clue that glucose somehow has a hold over those regulatory genes, or it controls those regulatory genes, okay, to make sure that the lactose metabolizing enzymes are not produced. Hope you understood. Now, there are some inducers known as gratuitous inducers. What are these? These are structural analogues of lactose which can induce the lac operon but are not substrate for beta galactosidase. Okay, the example is isopropyl thiogalactoside. Okay, this can be asked as an MCQ. Okay, so we also have many concepts working in this repression-derepression mechanism. As one, the very well studied is lac operon concept in E. coli. Okay, and in human beings it is induction of tryptophan pyrrolase by tryptophan. Okay, transaminases are induced by glucocorticoids. Okay, ELA synthase helps in heme synthesis. Okay, they are induced by barbiturates. Glucuronyl transferase are also induced by barbiturates. Okay, this is the example for tryptophan operon. Like how you had lac operon for lactose, you also have tryptophan operon in E. coli, where tryp, R, P, O and A are the regulatory elements. E, D, C, B, A are the structural genes which can produce polycystonic mRNA, provided the permission is given by regulatory elements, provided there is no inhibitory factors acting on these regulatory elements. So before we move on to the regulation of gene expression in eukaryotes, I will try to play a video.
Hey everyone, the Vikings Race here, so let's talk about the lac operon. The lac operon is the best studied model for understanding the regulation of gene expression. The genomic DNA of bacteria has many genes. Genes, by the process of transcription and translation, forms a protein. Both transcription and translation are highly energy intensive, hence only those genes that are required for homeostasis are continuously expressed. This type of expression is known as constitutive expression. Most of the genes are controlled by the extracellular signals, which are usually molecules in the growth medium. The signals that positively regulate the gene expression are called activators, while the signals that negatively regulate gene expression are called repressors. Both activator and repressor are the DNA binding proteins that regulate the gene expression. The DNA sequence at which the repressor binds is called an operator, while the DNA sequence at which the activator binds is called activator binding site. Now let's talk about the lac operon. An operon is a functional unit of DNA containing a cluster of genes under a single promoter. The lac operon has three genes, LAC-Z, LAC-Y, and LAC-A. The LAC-Z gene encodes beta-galactosase enzyme. This breaks lactose into glucose and galactose. The LAC-Y gene encodes lactose permease enzyme. This enzyme transports lactose inside the cell. The LAC-A gene encodes thiogalactoside transacetylase enzyme, and it helps in the removal of toxic thiogalactosides, which are also transported by the LAC-Y. The LAC-I encodes a repressor that binds the operator and represses the lac operon. Let's see how the lac operon functions. In absence of lactose, the operator is occupied by the lac repressor. This ultimately shuts down the lac operon genes. Basal level transcription. Now, sometimes in the absence of lactose, the repressor may fall off from the operator, allowing the transcription of lack genes. This occurs because the binding of repressor with the operator is not permanent. Because of basal level transcription, there is always some amount of lactose permease, beta-galactosidase, and thiogalactoside transacetylase enzyme present in the cell. When lactose is present, lactose enters the cell with the help of lactose permease. Remember, lactose permease is present because of the basal level transcription of the lac genes. The lactose that enters the cell binds to the repressor and releases the repressor from the operator. As the operator is free, the RNA polymerase can now bind the promoter and initiate the transcription of lac genes. Now, as lac genes are continuously transcribed, more amount of lactose permease, beta-galactosidase, and thiogalactoside transacetylase enzymes are formed. The lactose permease further allows more amount of lactose to enter the cell. The lactose that enters the cell is hydrolyzed to glucose and galactose by beta-galactosidase enzyme. Glucose and galactose are used as an energy source. The role of CAP protein in lac operon. CAP stands for catabolite activated protein. CAP binds CAMP, hence it is also known as CAMP receptor protein. When the glucose levels in the cell decreases, the level of cyclic AMP in the cell increases. The CAMP binds CAP protein and activates it. The CAMP-CAP complex then binds to the CAP site and interacts with the C-terminal domain of the alpha subunit of RNA polymerase enzyme. This increases the rate of transcription of lac genes.
Hope you understood. Okay, gratuitous inducer, right? Okay, there are some structural analogues of lactose, okay, which resemble the structure of lactose, which can induce the lac operon. Okay, that means it can have influence on the regulatory genes, which can then guide the structural genes to produce the enzymes of lactose metabolism, but they cannot act as substrates for beta-galactosidase. Okay, such substances are known as gratuitous inducers, and the example is isopropyl thiogalactoside. Now, moving on to the regulation of gene expression in eukaryotes. In eukaryotes, the expression starts at DNA level itself. Okay, till now, while we were studying the regulation of gene expression in prokaryotes, we saw that it is mainly at the level of transcription, okay, which affects the synthesis of protein, that is translation also. But in case of eukaryotes, it starts at DNA level itself, okay, and ends with the enzyme, that is the last product, okay, which is catalyzing a particular chemical reaction. Okay, so various, as I told you, the regulation of expression of gene in eukaryotes is complex because it is highly influenced by various signaling molecules, environmental signals, as well as regulatory proteins. It was very simple with just the regulatory proteins, the inducers and repressors, okay, for the eukaryotes to regulate, for the prokaryotes to regulate. In case of eukaryotes, it is just one of the elements of regulation. So it is highly regulated by signaling molecules and environmental signals as well. So the levels of gene expression in eukaryotes can be controlled at 5 different stages. That is, at DNA level itself, when it is undergoing replication, you can have a regulation. I think you would have studied regulation of the DNA metabolism at replication and transcription and translation. Okay, It can also be controlled at the level of transcription or at the level of synthesis of mature mRNA, that is post-transcriptional modifications. It can also be independently regulated at the level of translation. Okay, as soon as the RNA is formed from the structural genes, it used to translate into protein in case of prokaryotes. But here again, even at the level of translation, there can be independent regulation. Okay, even after the protein is formed, it can still be regulated. Okay, so post-translational modifications can also be regulated. Okay, now there are nearly 8 to 10 mechanisms of regulation of gene expression in eukaryotes. We will look into the main ones. Okay, so the genes are altered by gene amplification, gene rearrangement, gene loss and methylation of DNA, which is nothing but chemical modification of DNA. What do you mean by gene amplification? The expression of a gene is increased several fold. Okay, it is commonly observed during the developmental stage when the cell division is excessive, when the growth rates are excessive. Okay, now it also has a therapeutic implication because methotrexate, which is commonly used as an anti-cancer drug, acts by inhibiting the enzyme dihydrofolate reductase and thereby it interferes with the cellular metabolism. Okay, now the malignant cells or the cancerous cells which should have been the target for the methotrexate will develop resistance to this drug over a span of time. Okay, initially through this mechanism methotrexate can control the proliferation of malignant cells. Okay, like how the bacterial cells can develop resistance to the antibiotics, similarly even the malignant cells can also develop resistance to the anti-cancer drugs. Okay, through one of the mechanism known as gene amplification. They may develop gene drug resistance to the long-term administration of methotrexate by amplifying the genes coding for dihydrofolate reductase. Okay, then you have transcriptional regulation. It is a major method of regulation in eukaryotes. Okay, which is manifested at two different levels, that is chromosomal packaging and individual gene regulation. So the transcriptional factors, okay, can be enhancers, that is which positively influence the regulation, or it can also be negatively regulating proteins. Okay, so it binds to the protein promoter side and it can influence the transcription of RNA. Okay, so it can get incorporated into the RNA chain which is formed. Okay, so this is binding of transcriptional factors to the promoter and enhancer regions to form a loop. Okay, now post-transcriptional regulation. These mechanisms exert the regulation after the mRNA has been produced. Okay, so this is much more extensive in eukaryotes than prokaryotes because it's almost at the it stops at the level of transcription in case of prokaryotes. Okay, there are two major levels at this post-transcriptional level also. How it does is by alternative splicing or by regulating the RNA stability. Let me explain you what is this alternative splicing is. What do you mean by splicing? Okay, before discussing what splicing is, you need to know what introns and exons are. Exons are the expressed part of the genes, right? Introns are the intervening sequences which do not have any specific function. Okay, once the mRNA transcript has been produced from DNA via transcription, okay, we don't want to continue including the non-functional parts also in all the generations of DNA, RNA and protein. So at this level we do remove the non-functioning parts. We only take the actively functioning parts. So the introns or the intervening sequences are removed. Okay, and the exons which are lying in between the introns are joined together by a process known as splicing. Okay, so even those exons are actually different units, that is Systronically, their function is different with respect to each of the genes. Okay. Now, even in this splicing, okay, suppose a gene contains or mRNA transcript contains, for example, 10 cistrons. Okay. We need not express all the 10 cistrons of this mRNA in all the cells all the time. That is what I explained you initially. Okay. Maybe only some 8 out of these 10 cistrons are required for the organ A to function. Okay. So only those are expressed, the other 2 are suppressed. Okay. Maybe some organ B requires functioning of only 5 of those 10 exons. Okay. It is only those 5 which are expressed there, the other 5 are not expressed. Okay. So alternative splicing is one such mechanism, okay, which helps in individual tissues to perform their own functions. Now, translational regulation. This affects the initiation step itself, wherein the initiation factors, especially the eukaryotic initiation factor 2 is affected. Okay, it is involved in this regulatory mechanism. Okay, the action of this factor is inhibited when it is phosphorylated by a protein kinase. Like how you have the example of the enzymes of glycogen metabolism getting phosphorylated or dephosphorylated. Okay, to either stimulate or suppress the glycogen metabolism. You also have phosphorylation dephosphorylation process influencing the initiation factor. So when it is phosphorylated, kinases are the enzyme which help in transferring the phosphate group, right? When this protein kinase transfers the phosphate group to this factor, it gets phosphorylated, and in phosphorylated state it gets inhibited. Okay, so this is how at the level of translation the genes can be regulated. Now, post-translational controls will act only after the protein has been produced. Let me play you a video again.
Humans share over 99% of their DNA. That means that if you looked at any two people and you looked at their DNA sequences, 99% of the sequence would be exactly the same. And yet there's tremendous variation in phenotypes in the human population. Similarly, chimpanzees and humans, as two different species, still share over 98% of their DNA. Yet again, their phenotypes are quite different and they're separate species. In order to understand how this can be possible, and to better understand the process of cell specialization, we're going to take a close look at eukaryotic gene regulation. And we're going to focus on mechanisms that help control gene expression, particularly at the transcriptional level. So a quick review of gene expression. Remember, gene expression is turning on or off genes. And it consists of 2 major steps. To get a gene into a protein, we first have to transcribe the DNA into mRNA. That's the first stage of gene expression. Then we've got to translate the mRNA into a polypeptide, which makes up a protein, which makes up a trait. In this particular video, we're going to focus again on mechanisms of transcriptional control. So the first method is DNA packing into chromosomes. If you take a look at a chromosome, it's actually quite complex at the level of DNA. So let's start over here. Here's DNA, our double nucleus. In order to get DNA to fit nice and neatly inside the nucleus, this DNA is going to wind around histone proteins, like this. And it looks like these on the screen. The winding of DNA around the histone proteins forms a structure called a nucleosome. The nucleosomes then wind into a bigger helix, and then that helix winds into a supercoil, and then the supercoil loops and folds, and that's actually what makes up a chromosome. So a simple picture of a chromosome may lead you to think that the structure is pretty simple, but there's multiple levels of DNA packing. And this is important to transcription because in order for RNA polymerase to transcribe DNA, DNA into mRNA. Another method of transcriptional regulation is something we're pretty familiar with, transcription factors. Here we've got a DNA strand. The gene is right here. This is the part of the DNA where we'll actually code for a protein. Before the gene is a sequence of DNA called a promoter. We know that the RNA polymerase has to bind to the promoter in order to start transcription. But then, further upstream from the DNA promoter and gene are these sequences called enhancers. Enhancers will get bound by transcription factors. And the job of transcription factors is to help RNA polymerase start transcription. So here's what it looks like if the gene is going to get transcribed. Here we can see that the transcription factors are bound to the enhancer region, and in doing so, they have helped RNA polymerase bind to the promoter. Now RNA polymerase can start transcribing the gene. So to sum that up, RNA polymerase is the one transcribing DNA into RNA. The promoter is this green region that the RNA polymerase has to bind, and the promoter is DNA. The enhancer is another area of DNA, often quite far away from the gene, and that's what the transcription factors bind to. And the transcription factors are these purple and green molecules. And it's important to note, you're going to want to add this to your annotation, the transcription factors can activate or repress transcription. In this particular diagram, we are seeing activators, because they're helping activate the RNA polymerase. But there are other types that will repress RNA polymerase's activity and turn things off. Another type of control is our alternative splicing. We're familiar with this as well. Remember that in a DNA gene, not all of the sequences will end up in the final RNA. So the DNA gets transcribed into RNA, but only the exons, 1, 2, 3, 4, and 5, end up in the final RNA molecule. The introns, these non-coding regions, get cut out and discarded. And the cool thing about splicing is that you can splice the exons together in different ways. For this mRNA molecule, we splice together 1, 2, 3, and 5 to get a particular mRNA molecule. In this one, we splice together 1, 2, 4, and 5. So the cool thing about this is with one DNA sequence, we can get multiple RNAs, which means multiple proteins. And this helps explain some of the variation in phenotypes that we see within humans or with humans compared to chimpanzees. Because even if two different people have the same DNA sequence, we might get different traits depending on how the RNA gets spliced together. Another method of controlling transcription happens slightly after transcription, in fact. Here's our mRNA molecule. You can see it's got its cap. But eventually the mRNA molecule is going to get broken down or degraded. And when that happens is key. Because the lifespan of mRNA determines how much protein is made. Eventually the mRNA will get broken down by enzymes and RNA, but depending on how long it takes to get broken down, the mRNA molecule could last a few hours or many, many weeks. And the longer it lasts, the more protein is going to be made. So if a cell needs a lot of a particular type of protein, it won't degrade the mRNA right away. If the cell only needs a little bit of protein or the environment changes and it doesn't need that protein anymore, it can respond by breaking down the mRNA into little pieces so that we don't get any more protein. And there's a specific mechanism involved in degrading mRNA. It's called RNA interference. This is a hot topic in science right now. Remember that only 2% of the human genome consists of genes that code for proteins. You get 98% of your DNA that's not producing proteins, that's not contributing directly to traits. For a long time, scientists just thought that 90% was junk, but it turns out that it's not really junk. In fact, some of it is transcribed into tiny little RNA molecules called microRNAs. And here's what those microRNAs do. A microRNA, here's the short little RNA, will get together with some proteins and it will bind to we have stopped translation of the mRNA into a protein. So this is a major mechanism for controlling gene expression. And this also contributes to variation among species. And that's all for today. Please make sure you're notes of all the key components and don't forget to do both.
So I hope it is clear for you now. Okay, if you do have any doubts, please post it so that I can explain those concepts again. Yes, you do not have any doubts? That either means you have fully understood or not understood at all. So the lack operon concept, regulation of gene expression in eukaryotes or prokaryotes can itself be a main question, or lack operon concept can itself be a 5-mark question or 3-mark question for you, and it can also be included in MCQs. So they can ask you the various levels of regulation of gene expression either in prokaryotes or eukaryotes, and various methods by which they are regulated. Okay, this looks simple when you are just listening. Only when you have to put down on papers you get the real problem. So please go back and read the textbooks today itself. Okay, make a notes of your own. I'll be displaying you the assignment questions. Try to answer them and try to manage the time as well. Any doubts? Okay, if you do not have any doubts, let me ask you a few questions. Name the regulatory genes of lack operon. Only the regulatory genes, not the structural genes. Okay, name the regulatory genes of lack operon. No answers? You guys are not listening to class anymore? Okay, very good. I, P and O genes. I for the regulatory gene, P for promoter side and O for operator side. Now what about structural genes? What are the structural genes of lack operon? Yes, Z, Y and A. Okay. What are the enzymes synthesized by each of these structural genes? What enzyme is synthesized by Z, what enzyme by Y and what enzyme by A? Okay, Z for beta galactosidase, very good. Y for permease, very good. And A for transacetylase, very good. Now this beta galactosidase is the enzyme which actually splits down lactose to galactose and glucose. What is the action of permease? Permease helps in the entry of lactose into that cell, permeation, okay. And transacetylase probably is involved in taking care of the inhibitors which are also synthesized together with permease so that it inactivates them. Okay. Now, how can you define operon? What is operon? What is operon? Okay, it is a coordinated unit of gene expression. Very good. So, operon refers to a cluster of genes or a set of genes, okay, which act in a coordinated fashion. Okay, if these genes have the effect on enzymes of lactose metabolism, it is called lac operon. Similarly, you have tryptophan operon and such examples. Okay, very good. What are constitutive genes? What are constitutive genes? What do you mean by constitutively expressed? Correct. Those genes which are constantly expressed, okay, are called constitutive genes. They do not require regulation, or rather they are not regulated. They keep on performing their duties as and when it is required. Okay. So, in contrast to constitutive genes, you have inducible genes. What are inducible genes? What are inducible genes? Correct. These are the genes which are regulated or which requires a stimulus for it to express. Okay, which requires inducer for it to express. Also, only then it is expressed. These are called inducible genes. Any doubts? I hope you understood the concept of repression and de-repression. I will try to clarify it once again. Repression is nothing but negative regulation. So, with respect to lac operon, normally, okay, when there is in the absence of lactose, okay, the genes are responsible for the metabolism of enzymes or the synthesis of enzymes required for lactose metabolism are repressed or suppressed by the help of lac repressor protein, which is produced by the regulatory gene I. Okay. In presence of lactose, when you supply lactose to those cells, okay, the repressor protein, instead of going and binding to the operator site and influencing the promoter region as well, okay, it goes and binds the lactose molecule. So, there is no pressure or there is no regulatory element sitting on the operator gene anymore, okay. So, that the neighboring promoter region is free to act. How does it act? It acts by binding the RNA polymerase. It acts by guiding the RNA polymerase to the transcription initiation site, so that the structural genes Z, Y and A produce the mRNA, which is produced as a single mRNA, okay, and each cistron of these mRNA will produce 3 different enzymes. So, this is known as polycistronic mRNA, and the 3 enzymes produced are beta galactosidase, permease and transacetylase. Okay. Thank you. Please answer the assignment questions, and if you still have doubts, you can ask me in WhatsApp. Thank you.
one of the abdominal organs in our body, that is in relation to the spleen. So this has to be learned under the following headings, as you must be aware by this time. So how exactly you are supposed to write to the this particular topic. Now it has to be under these following headings, as you can make out over here. As you have to describe the spleen by defining it, then location or situation where exactly the organ, this particular organ is located with respect to the abdominal cavity. Then a word about development if it is asked for. Then obviously the external features of this organ particularly and its relation and blood supply as well. And last but not the least would be the applied aspect related to the spleen. So this is how you are supposed to learn about the spleen. Now we will go ahead with this organ spleen. So as usual in as an introductory part. So as an introductory note, so what are we supposed to know with respect to the spleen? So now spleen even has a synonym terms or name, because these terms you come across when some of the relations when they are described with respect to the spleen or in relation to the spleen, then these terms like the spleen in Greek term also called as spleen or related as spleen, and in Latin term as a lean. So these are the terminologies that you will come across while you are reading, so as to be like the ligament or any of the relation that you will come to know as the particular topic now. Now, before we look into define what exactly the spleen, now briefly about the functional aspect of the spleen. So as you can make out here, so this is the location. We will see that again in the next slide. So this is where, as you can make out from the diagram, the abdominal cavity, you can make out the location of the spleen with respect to the stomach. So obviously, mainly particularly the fundus part of the stomach, and you can make out over here being situated in the left hypochondriac region. This is slightly magnified version of this particular image. Now the spleen is considered to be like an organ which filters the blood. So it will filter the blood from basically two aspects. It filters in two aspects. One, it filters the worn out or the older RBCs to be removed from the circulation, and the other one, it also filters the blood from the microbial agents. So that is how the spleen functions. Apart from that, no wonder it also maintains the immunity of the particular individual by producing specifically this antibody or the immunoglobulin type is M type of immunoglobulin. You might be aware that immunoglobulins have been divided into 5 groups as GAMED, so in that order. So immunoglobulin M is primarily produced by the spleen, providing immunity to the particular individual. Apart from that, spleen also helps in producing lymphocytes, no doubt in adult after the birth, but the newborn individual or in a fetus particularly, it also helps in producing or synthesizing the RBCs. That is how the spleen functions. Apart from that, also one more function would be it stores the RBCs and whenever it is required, depending upon the situation, so it will be released into the circulation. So these are about the 3 to 4 functional aspect of the spleen, so that performs in our body. Now we look into where exactly the spleen location is. So whenever with respect to the examination point of view, so you have to describe the location of the exact location of the spleen. So that is described to be or spleen to be located in the left hypochondriac region. So as you can make out here, this is like we are seeing the back of a particular individual. So this image is nothing but you're seeing from the back. So this being the left side and the right side. Now more particularly, so location would be a situation would be left hypochondriac region. I hope even you must be or you will be aware by this time how the abdominal cavity has been divided into 9 quadrants by 2 vertical and 2 horizontal lines. So when you are aware of that, so we say that the left hypochondriac region is the exact location of the spleen. Whereas to be more specific, as you can make out here, so this dotted line represents the mid-axillary line. So slightly behind the mid-axillary line. So in the left hypochondriac region location, slightly behind the mid-axillary line. So that is how it is described, and it can also be described with respect to the ribs over here. So particularly the spleen location would be in relation to the 9th, 10th and the 11th ribs over the particular costal region, right in the left hypochondriac region. Whereas if we look into the now this is how the image slightly magnified, so that you can have a better visualization. So like you are looking from the back, so left hypochondriac region, and this representing the mid-axillary line. So again, you can make out here. So especially when we say with respect to the ribs, that is as 9, 10 and 9, 10 and the 11th rib, which is in relation to the spleen. More particularly, the 10th rib is considered to be like the long axis, parallel to the long axis of the spleen. So that is how they describe with respect to the ribs as well. This is what I said about. So long axis of the spleen lies almost in parallel to the or in relation to the 10th rib. So of course, even being very close approximation to the diaphragm as well, so it moves as its motility with respect or movement will be noted or can be recorded with respect to the respiration. Whereas now with respect to the shape and size and the color of the spleen, so it has been described as to be depending on the percentage of the people, so which is seen, it is said to be spleen as said to be in about 3 different sizes. So as it has been mentioned over in this diagram, we can make out in the diagram. So now this is how it is considered to be the wedge-shaped spleen, and this is to be tetrahedral shape, and this is more or less like a triangular in shape. So that is how they describe the size of the shape of the spleen. Now more profoundly or more in more percentage of people, so the spleen is considered to be a wedge-shaped organ, which is more or less has a purple in color or appearance. It appears to be more purple in color. So wedge-shaped organ seen almost in 44% of the people. So it is considered to be or it is seen as a wedge-shaped organ. Now to be, if you, to define the spleen, it would be considered as the largest lymphoid organ in our body. So that is how we define the spleen in one sentence to be as the largest lymphoid organ in our body. Now, more, in a large percent of the people, almost in 44% of the people, it will be wedge-shaped, as you can make out over there. This is how the statistics based upon the shape of the spleen in different groups of people. So 44% of the people it will be wedge-shaped, whereas around almost 42% of the people it will be considered to be tetrahedral in shape. So this one, whereas in about close to 15% of the people, so it will be considered to be triangular in appearance or the shape. So this is how they describe the shape of the spleen to be wedge or tetrahedral or triangular in shape. Whereas again, like any other organ in our body, so we have to even be aware of the dimensional part of the spleen as well. So as you can make out, it is considered to be, our spleen is defined with respect to dimension wise, it is defined as to be 1 inch thicker, it is just 1 inch thicker, it will be 3 inches broader, so it will be 1 inch thicker, it will be 3 inches wider or broader, 3 inches broader. Whereas lengthwise it will be almost 5 inches longer, so that is all, and weighs almost about 7 pounds in weight. So that is how they describe the dimensional aspect of the spleen, and it is quite if you can see over here, it is all of them are odd number, described in odd number as 1, 3, 5 and 7. So that is they use a dictum to denote those dimensional views. So it is called as the Harris dictum. So it is called as the Harris dictum in an odd number fashion. So 1, 3, 5, 7, whereas 9, 10 and 11. So 9 to 11, that is that denotes the relationship of the spleen with respect to the ribs, as you can make out in the diagram. So 9 to 10 and 11th rib. So in relation, since it is in close relation to these 3 ribs, so that also has been included. So that is called as the Harris dictum. So see that you remember that 1, 3, 5, 7, 9 to 11. So it is 1 inch thicker, it is a 3 inch wider or broader, 5 inches longer, and 7 ounce, weighs almost 7 ounce, and related to the these 3 ribs, 9, 10, and 11 ribs. So that is called as the Harris dictum. Whereas after knowing the axial features of the spleen, so now we'll see after knowing the dimensions of the spleen, now we'll go through the excellent aspects of the spleen. So what exactly a normal spleen, considering that wedge-shaped organ, so see, which is almost 44% of the people, in relation to that. So what are the excellent features that every spleen projects? Now, each of the, every spleen should be, we should be able to make out having 2 ends, 3 borders, 2 surfaces, and 2 angles. This is how the excellent features can be explained under these headings as well. Now we'll take up each of the features, then we'll go through the slight details about these excellent features. Now first, if you take up the ends part of the excellent feature, it has 2 ends, as we can make out. 2 ends are considered to be the anterior end, to be simple as anterior end and the posterior end. So now this is considered to be the anterior end of the spleen, and this is the posterior end of the spleen. But it also, depending upon the author, so it has been again, there is a synonym terms for the ends as well. Anterior end can also be called as a lateral end or the lower pole of the spleen. So this can be anterior end or the lower pole or the lateral end of the spleen. So this can also be called as, apart from being the posterior end, it can also be called as the medial end and the upper pole of the spleen. So that is how they can use even the synonym terms as well. Whereas now first, if we take up the first one, the anterior end of the spleen over here. The anterior end of the spleen it doesn't appear to be like an end, but when we read through the word as an end, it comes to be like more like a narrow part of a particular region or a structure. But here it is unlike over here. The anterior end appears to be more like a border. So that is how we have to be aware of that particular part. So anterior end appears to be more like a border. So otherwise, this is the anterior end. Now the direction, it is said to be like directed downwards, forwards, and to the left side. So that is how the direction downwards, forwards, and to the left side. So that you have to mention with respect to the anterior end. Now it is related to, very importantly, the anterior end is related to these 2 structures. One is called as a left pollic flexure. So left pollic flexure. The other one is a ligament called phrenicopollic ligament. Now, what are these 2 structures? Then we'll see with the diagram. Then you will get a better imagination. Now the anterior end, so it is related to, one would be the left pollic flexure. So this is again a magnified view, just to make you understand. The posterior end, this is the anterior end, looks more like a border. Entire is the anterior end of the spleen. Whereas comparatively, border posterior end is quite narrow and blunt. So that is how it can make out. Now this image is mainly to be aware, to be more familiar with the surfaces. We'll see that later. So this being the 2 surfaces. Now, what exactly the anterior end was related to, as we have seen, related to 2 structures. One is the left pollic flexure, as you can make out from the diagram. So this being the liver. Now you can make out the stomach outline, and behind the stomach, what you can make out in relation to the funding part, you can see the impression of the spleen over here. Now, if you closely observe, the rest of the GI tract is also been shown over here. So we'll stick on with the large intestine, the ascending colon, the transverse. Then the descending colon. Now with respect to, this is called as a right pollic flexure over here, and this is called as the bend. Second bend is a left pollic flexure. So spleen is related, anterior end of the spleen is related to the left pollic flexure. So this is in relation to the spleen. That is especially the anterior end of the spleen is in approximation to the left pollic flexure. This bend that you are seeing over there. Apart from that, the other ligament we saw that is a phrenicocolic ligament. Now this is again another image where you can make out the spleen can be seen and the contact of the pancreas over here. Now this is the diaphragm which has been shown in the background. So this is the diaphragm. Now between the colon, again you can even make out the left colic flexure, the bend over here, transverse colon, continuing as a descending colon. This bend is called left colic flexure. Now between this left colic flexure and there is a connection between the left colic flexure and the diaphragm. That is called as a phrenicocolic. That's what is called as phrenica. That is phreno related to the diaphragm. Colic, that is the left colic flexure. So phrenicocolic ligament. So that is also in close contact with the anterior end of the spleen. So two things are in relation to the anterior end of the spleen. Left colic flexure, this one, and the phrenicocolic ligament. So those are the two important relations to the anterior end. Now you see, when if you move on to the posterior end, it is as I had told you earlier. So compared to the anterior end, posterior end is quite blunter and narrower. So that is you can clearly make out the difference with respect to considering that spleen to be a wedge-shaped organ. So these are related with respect to that shape of the spleen. Now this is like a direction-wise. This was directed downward, forwards, and to the left. Now this will be directed upwards, backwards, and medially. So that is the direction of the posterior end or the posterior pole, also called as the medial end of the upper pole of the spleen. Now this is related to just like the anterior end which was related earlier to the ligament and the part of the colon. Now this is more approximated to the upper pole of the left kidney. Not simply remember it as it is more approximation to the left kidney. Since the spleen is in the left hypochondriac region, so obvious would be this should be in relation to the left kidney. So that is about the two ends of the spleen. Whereas now if you look into the borders, we said that there are three borders with the spleen, considering that the spleen to be a wedge-shaped organ, which is seen in majority of the people, like 44% of group of people. Now this is the superior border. Then there are three borders. This is the superior border, and this is the inferior border, and the one between them, this region is called as the intermediate border. So that is how the three borders are described with respect to the spleen. Now if you take up the superior border, so this is the one, this superior border. Now this separates the two surfaces of the spleen. Now what you see here, this surface of the spleen is called visceral surface of the spleen. The one opposite to that is the diaphragmatic surface of the spleen. Now as you can make out here, this is the visceral surface of the spleen. Why visceral? Because it will come in contact. Next we will see the relations of the spleen. Now this is the surface that comes in contact with the different viscera. So that's why visceral surface. Whereas this is the one which is in contact with the diaphragm, dome of the diaphragm. So that's why it's called the diaphragmatic surface. So you can consider this one as the superior border. Separates the diaphragmatic surface from the visceral surface. That is how it separates. Now the most characteristic, one of the most striking or the characteristic feature on the superior border is, as you can make out over here, you can see the depression, the notch-like feature. So this is the characteristic feature of the spleen, especially now to differentiate from which is to be the superior to the inferior border. So whenever you are getting confused, so see that if the border displays shows or projects these notches, so that should be the superior border, unlike the other one should be the inferior border. So that is how you can differentiate the spleen from superior. the borders of the spleen from superior to the inferior border. Now that is what it means over here. So now why these notches? Now these notches are seen only on the seen on the superior border of the spleen. Now it has its own developmental or embryological reason as well, because spleen during the developmental process, spleen develops from masses of mesenchymal tissue, especially in the dorsal mesogastrium, that region. So now the spleen as a whole is formed by union or fusion of different masses of mesenchymal tissue during the developmental process. So that denotes that the notches being present over here. So the notches represents that spleen does not develop as a whole, as a one particular mass of mesenchymal tissue, but develops as a group of or multiple masses of mesenchymal tissue, which in turn fuse to form one particular organ, that is the spleen. So that is what represents the notch being present on the superior border. If you see the inferior border, so quite smoother. So as you can make out, usually what other thing would be superior border shows slightly thinner compared to the inferior border. which appears to be slightly rounded. So that now this border again, inferior border, again separates the visceral surface from the diaphragmatic surface, and this border in relation to the 11th rib, it will be more closer proximity to the 11th rib. So that is how we can describe in relation to the inferior border. So much of the feature, much of the importance should be with the superior border, bearing the notches over here. So that differentiates both of the borders. But the third border is this one, the middle or the intermediate border. So this one. So wherein you can make out of the hilum, the gap or the opening wherein structures enter and come out of the splenic tissue. So that represents the intermediate border over here. So rounded separates these two. Now this is the intermediate border, this region. So it separates the two areas over here. So this upper and the lower area, which is called as the gastric impression and the renal impression. And we'll see that with respect to the relations of the visceral relations of the spleen. So that is how the three borders can be remembered apart from the two ends. Now we'll see the two surfaces of the spleen. Now two surfaces of the spleen are, as we are already aware. So this is the diaphragmatic surface of the spleen, and which is in contact with the diaphragm, obviously, and this is the visceral surface of the spleen. So these are the two basic surfaces of the spleen. Now diaphragmatic surface, as is very much evident, it does not show any features over here, appears to be quite convex and smooth over here, which represents the diaphragmatic surface. Obviously this should be directed upwards, backwards, and to the left. With respect to the position of the spleen being present in the left hypochondrial, so this direction can be mentioned as upwards, backwards, and to the left. That should be the direction of the diaphragmatic surface. And if you see the visceral surface appears to be shows slight depressed areas or concavity. So that's why they use this term of the concavity. Visceral surface does not appear to be uniform in appearance, just like what we saw. Unlike what we saw with respect to diaphragmatic surface, appears to be uniform and convex, but here it shows slight depressions which are called diffuse concave like appearance over here. So that's why that differentiates the diaphragmatic from the visceral surface. Now what is important is with respect to visceral surface, it shows it is related to four important viscera surrounding the spleen. So those would be in case with respect to the stomach over here or gastric impression or the gastric area, with respect to the left kidney, so renal area, and with respect to anterior and over here, so it is related to the left colic flexure, and with respect to the hilum over here related to the tail of the pancreas. So that's why there are three impressions or areas with respect to the visceral surface of the spleen, has to be gastric, renal, colic, and pancreatic. So we'll see that again in the visual relations, then we'll get a better idea. So that is about the surfaces. Now same as I said, so with respect to if you go into slighter detail of the visual surface here, so this area between the superior border and the intermediate border, so this area, depressed area, is called as it is called as the gastric area or the gastric impression, because it is related to the funding part of the stomach, whereas the second area would be the renal impression. So between the intermediate border or the middle border and the inferior border, so this area is called as related to the left kidney, so that's why it's called as the renal area or renal impression. Whereas just behind the anterior end of the anterior end, as you can make out over here, just behind the anterior end. This is the anterior end, which we are already familiar. Anterior end appears more like a border. So just behind the anterior end, this area is called the colic impression, where it is in contact or where it is in approximation with the left colic flexure. Whereas the fourth area impression would be, again, as you can see over here, around the hilum, this region. So you can make out around the hilar region or the hilum of the kidney, hilum of the spleen. So you can make out the tail of the pancreas will be in contact over here. That's why it is called the pancreatic impression. So there are four viscera which are related to the spleen. One, gastric area between the superior border and the intermediate border. Renal area, that is related to the left kidney between the intermediate border and the inferior border, and just behind the anterior end of the spleen, so it will be related to the left colic flexure, and just in front of the hilum, or you can say between the colic impression and the hilum. So this area is related to the tail of the pancreas. That's why it's called as the pancreatic impression. viscera are in contact with the visceral surface of the spleen. So after that, one of the again fourth feature, feature related to the spleen would be called as two angles. Now it depends on, some of the authors from clinical point of view, they describe the spleen to be having two angles. One has the anterobasal angle, so this one anterobasal angle, the other would be the posterobasal angle. Now what exactly is the anterobasal angle? The point of the junction between the superior border and the anterior end. So that is what the junction between the superior border with the anterior end of the spleen. So this particular region of the spleen or part of the spleen is called as the anterior angle, otherwise also called as, more specifically called as the anterobasal angle, also otherwise called as the clinical angle of the spleen. So anterobasal is also called as the clinical angle of the spleen. Why clinical angle? Because whenever, if at all, whatever the cause, if the spleen enlarges, so when the doctor, as a physician, when he tries to palpate the enlargement of the spleen, to know about the enlargement of the spleen, so the first part of the spleen that comes in contact with the physician's fingers would be this part of the spleen. So that's why this anterobasal angle is also called as the clinical angle of the spleen. Whereas the second angle is the posterobasal angle between the inferior border and the anterior end again. So this is the posterobasal angle. So these are the two angles which are again equally important, which has been described with the spleen. Now we look into the relations of the spleen. Now relations of the spleen has been again categorized for better understanding into two groups, as to peritoneal relation and the visceral relation. So anyway, visceral relation, we are almost familiar with what are the viscera that are in relation to the spleen, so that won't be much, won't take much of time to even recollect in examination. But with respect to the peritoneal relation, so what are the peritoneal relation? That is in close approximation to this organ, that is the largest lymphoid organ in our body, is the spleen. Now, two folds of the peritoneum, they are in very close approximation or in relation to the spleen over here. So one is called as the gastrosplenic ligament, another one is lieno-renal ligament. Now, as you can make out a cross-sectional diagram, a sectional diagram of the hypochondriac region, left hypochondriac region, you can make out this is a spleen over here, and you can see the left kidney, and equally, you can even simultaneously see the end of the pancreas, the tail of the pancreas can also be seen, and you can even see the cross section of the stomach as well. Now, what are the two ligaments? Gastrosplenic. Now, between the stomach, especially the greater curvature of the stomach. So now, stomach has less than greater curvature. Upper part of the greater curvature of the stomach and the spleen. So this fold of the peritoneum that you are seeing here, this entire fold of the peritoneum is called as a gastrosplenic ligament. Whereas, same way, now there is one more fold with respect to or between the spleen as well as the left kidney. So this fold of the peritoneum is called as lieno-renal. So lieno, as we are aware already, the synonym term of the spleen is also called as the lean apart from spleen. So lieno-renal, between the spleen and the left kidney, mind you, so left kidney. So this ligament is called as lieno-renal ligament. This between the stomach and the spleen also called as the gastrosplenic ligament. So those are the two peritoneal relation, two important peritoneal relation regarding or with respect to the folds of the peritoneum. So these two ligaments has to be mentioned again. Remember. Whereas now this is like specifically, as I told you earlier. So from where to what to what, or what part of the spleen to what part of the stomach is the ligament described. So it has been, as I said, so it is with respect to this ligament, gastrosplenic extends between the upper part of the greater curvature of the stomach to the hilum of the spleen. So that is how the gastrosplenic ligament extension can be mentioned. Now basically this consists of, as you can make out, a vessel running across over here considered to be consists of short gastric vessels. So are to be seen in this ligament. So along with even the developmental wise, again you can make out these structures represents the accessory spleens. Now what is accessory spleen? We'll see that in the coming slides again. So otherwise, so try to remember. So gastrosplenic ligament. So So consists of short cascade vessels. Also you can make out even some of the group of the lymph nodes as well as the accessory spleens. If at all, if in that particular region, if it due to developmental changes or developmental irregularity, I suppose. So in that way, you can even make out spleen tissues in the ligament as well. So those are called as accessory spleen. Whereas the lieno-renal, as I said, between the hilum of the spleen and the anterior surface of the left kidney. So that is how the lieno-renal ligament. So lieno-renal ligament between the hilum and the anterior surface of the left kidney. So that is the lieno-renal ligament. And what does this consist of? So this lieno-renal ligament consists, as you can make out, the tail of the pancreas can be seen, and you can even see one of the larger artery, that is the splenic artery. So this is splenic artery or splenic vessel, generalized term, artery plus splenic vein. So collectively called as a splenic vessel, and there will be a group of lymph nodes. This group of pancreatic splenic group of lymph nodes can also be included as a content in the lieno-renal ligament. So that is how the peritoneal relations of the spleen can be described. Whereas the visceral relation of the spleen, so which you are very much aware already. So nothing to I don't want to bother you too much. So we already seen what are the four viscera. So the gastric impression, renal impression, the left colic impression, and the pancreatic impression. So those are the four viscera, so which are in close approximation to the visceral surface. And one more to be remembered would be the diaphragmatic surface of the spleen. We already know that the diaphragmatic surface of the spleen is in approximation to the 3 ribs, that is 9, 10 and 11. So those are the relation with respect to peritoneal, with respect to spleen in the form of peritoneal and visceral relations. Now the blood supply of the spleen, blood supply primarily spleen is supplied by vessels which are called as the splenic vessels or splenic artery. So the chief source of blood supply to the spleen, as you can make out in the diagram over here, so which has a tortuous course, which is called, this artery is called as the splenic artery. So it is considered to be one of the largest branch of the celiac trunk. We'll see in the next topic. So in the branches of the celiac trunk, as you can make out, this is the celiac trunk or the artery. So this is giving the branch, this is the splenic artery. So it is a very tortuous course because tortuosity represents the movement of that spleen. Will be spleen engages especially with respect to breathing and respiration, and even depending upon the content of the contents in the stomach, depending on like distension of the stomach. So during those kind of activities, so obviously spleen being in contact with the stomach, close approximation to the stomach, so shows movement. So that's why to prevent, to make it more facilitative, so these artery, splenic artery appears to be more tortuous in course. So that is the chief artery, source of artery blood supply to the spleen. Whereas again, this, if you wish to remember, like depending upon the terminal branches. Now, if you drain the splenic artery, it ends as the terminal branches, as you can see over here. But depending upon where the splenic artery terminates, like if the terminal branches are long and rise away from the spleen, away from the hilum of the spleen, if the termination appears to be away from the hilum of the spleen and appears to be the terminal branches appears to be longer, so in that case, that spleen, can be classified as a diffuse type of spleen. And opposite to that, if the terminal branches are shorter and then appears to be in close approximation with the hilum or close to the hilum, so that would be considered to be the compact type of spleen. So that is how, depending upon the termination of the branches length and the location, has been described as a diffuse type and the compact type. Whereas venous drainage of the spleen, no doubt it is formed by one large vein, that is the splenic vein, so it runs with the splenic artery. But the formation of the splenic vein is based upon about almost 4 to 5 tributaries which emerge out from the spleen, and they fuse to form one large vein, that is the splenic vein. So as it runs almost behind the tail and the body of the pancreas, as you can make out this image, it has been shown in dotted line, representing that the splenic vein lies behind of the back or the posterior aspect of the pancreas, the tail and the body of the pancreas. So again, pancreas has its own parts, has to be having the head of the pancreas, the neck of the pancreas, the body, and the tail of the pancreas. This is considered the processing, that you will learn it later. Now this splenic vein formed by union of about 4 to 5 or 4 to 6 smaller tributaries. They join to form splenic vein, runs behind the tail and the body of the pancreas. Now till it comes in contact with the superior mesenteric vein over here. So when the splenic vein joins or fuses or joins with the superior mesenteric vein, it will run as a portal vein. One of the important vein concerned with respect to the GI tract. So that you will again learn in detail in the coming classes. So that is how the splenic vein can be described with respect to the pancreas, to be behind the pancreas. Same image as you can make out over here. The tributaries going to form the splenic vein. That's why it has been shown in the dotted line, to be suggesting that it is to be running behind the pancreas. So those are the that is how the splenic or the venous blood from the spleen drains out. Now the splenic vein again, as you can make out in the diagram, it has its own tributaries as well. So apart from being the about 4 to 6 smaller tributaries joining to form the splenic vein proper, it also receives the splenic vein also receives, as you can make out, short gastric vein. They are also draining over here. The left gastroepicloid vein also draining into the spleen, and inferior mesenteric vein also draining into the splenic vein apart from the pancreatic veins as well. So that is how we can mention even as a tributaries of the splenic vein. So can also be mentioned in that. So as we can make out in this image, so tributaries of the splenic vein can be described as short gastric vein, left gastroepicloid, pancreatic, and inferior mesenteric vein, which I showed earlier. So those are considered to be the tributaries and the venous drainage of the spleen. The lymphatic drainage. So now this being the largest lymphoid organ in our body, so whatever the blood, that is the tissue fluid, which is present within the spleen, because of spleen having multiple venous sinusoids, so the tissue fluid from the actual tissue of the spleen directly drains into the venous through these venous sinusoids. It drains into the splenic vein, whereas the lymphatics will be described with respect to spleen in regard to the other connective tissue of the spleen. So the connective tissue of the spleen can be in the form of capsule or trabecular or the peritoneum, which is in relation to the spleen. So with respect to these structures in and around the spleen, that is in the form of a capsule or the septae or the peritoneum, which is in and around the spleen, they have a lymphatic drainage. Now those lymph from those structures, that is in the surrounding in and around the spleen, they drain into this particular group of lymph nodes, that is pancreatico-renal lymph nodes. So that is around the pancreas and the spleen. So those group of lymph nodes, pancreatico-renal group of lymph nodes, drain the lymph from the structures in and around the spleen, but not from the actual splenic tissue, because that will drain directly the tissue fluid from there directly drains through the venous sinusoids into the veins which are present in and around the spleen. So that is also, this being the largest lymphoid organ, so that is the reason why it does not have a lymphatic drainage of its own except the structures in and around the spleen. That is about the arterial, venous, and the lymphatic. What about the nerve supply? So sympathetic, especially from the celiac plexus around one of the artery with respect to from the abdominal line, that we will see in the next chapter, next topic. So celiac artery has a plexus around that celiac plexus, especially from that plexus. The sympathetic nerves, they supply they have a vasomotor function with respect to the blood vessels, so which are supplying the spleen. So that is how the nerve supply to the spleen can be mentioned. Whereas a word about the segments of the spleen. Now the segments of the spleen, what happens is, now this is based upon the terminal branches of the spleen. As we have seen earlier, that the splenic artery ends ends close to the hilum, has been dividing into the terminal branches. Now those terminal branches also called as segmental branches. Now basically 2 to 3 terminal branches, usually 2 or maybe 3 terminal branches of the splenic artery. So they divide and enter into the spleen to supply the spleen. So if that region, in most of the people, I mean in most of the cases, if there are 2 terminal branches that divide the spleen into 2 segments, has to be superior and inferior segments. So that is how they divide it, as a superior segment of the spleen and the inferior segment of the spleen. Now this is like now between the 2 segments, if at all if there are 2 terminal branches, now between the 2 segments there will be a very thin avascular plane or avascular region or aspect of the spleen. Now this will help the surgeon, if at all, whatever cause or region, if at all, if the surgeon wishes to undergo, if the surgeon plans to do a splenectomy surgery on that particular individual, so he can go for the segmental resection of the spleen, either superior or inferior segmental resection, since there is a vascular plane and can be located and can be resected, depending on whatever the cause of the condition of that particular patient or the person. Now usually what happens is the avascular zone or the plane will be between the 2 segments or the vascular segment, and that will be can be mentioned with respect to the long axis of the spleen. There is the 10th rib along which is considered to be the long axis of the spleen. So that almost runs perpendicular to the avascular plane. So that also helps a doctor or a physician or surgeon to exactly locate the avascular zone of the spleen. So that is how they can that will be helpful in surgery if the surgeon plans. So that is about the segments of the spleen. A word briefly about the development of the spleen. Now spleen is said to be, as I told you earlier, the notches on the superior border of the spleen represents the developmental process or the procedure of the spleen. Spleen develops between the 2 layers. Now this might be aware of from this particular section view of the developmental diagram. So stomach and the liver and the spleen over here. This is called as the dorsal. This is called as the dorsal mesogastrium, and ventral mesogastrium is this part. Now spleen develops in the dorsal mesogastrium. So it's in the upper part of the dorsal mesogastrium. So how it develops a condensation of masses of mesenchymal tissues. Now those multiple masses of mesenchymal tissues, they fuse to form as an organ, as a whole. So that is how the notches represents on the superior border that we have seen earlier as an external features. So now those multiple masses of mesenchymal tissue, which in future leads to formation of a spleen as a whole. Now those masses are also they use a specific term for the called as splenenculi. So representing the multiple masses of splenic tissue that will join to form one large spleen. Now, with respect to development as well, a word about something called accessory spleen. So, since this is again in relation to the developmental process, because spleen develops by fusion of multiple masses of splenic tissue, that is splenenculi. So, every chances that accessory spleen, if at all, if those multiple mesenchymal masses, due to whatever condition or reason, if they fail to fuse to form one single splenic tissue or the spleen, so there are every chances that it might have an ectopic presence of splenic tissue other than the normal region. So that particular case or condition is called as to be an accessory spleen. Now usually accessory spleen, if at all if it happens in any particular individual, now that is more commonly seen in these in the ligaments, that is gastrosplenic, glenorenal. So you have seen already in this image, gastrosplenic, glenorenal ligament, and even in the greater momentum of the greater momentum with respect to the fold of the peritoneum, with respect to the greater curvature of the stomach. So even in that region also you can make out accessory splenic tissues. So that is called an accessory spleen. Now after aspect related to the spleen would be one as a palpatory. This is considered a very commonly used palpatory method, starting from the right iliac fossa that will be proceeded towards the left hypochondriac region. So that is how usually the palpation of the spleen as a procedure is followed, right? Starting from right iliac fossa, coming towards the mid, and then going towards the left hypochondriac region. So this is how the palpate procedure which is followed with respect to palpation of the spleen. palpation of spleen is normally in a normal person. It is very difficult to palpate in a normal individual unless the spleen enlarges more than 2 times. So only then the spleen becomes palpable. So remember that. So normally it is not palpable, being very safely located in the left hypochondrial region. So only if it becomes more than 2 times enlarges, only then it is palpable. And any enlargement of the spleen, if it happens, it will be coursing through this this path. So spleen, if it enlarges, it will be like it will be passing through this one, and it will be best fed with the massive splenomegaly, best fed even in the right hypochondrial, right iliac fossa as well. So that is how the massive splenomegaly can be even located. That is the reason why when they are palpating the spleen, so you have to start from right iliac fossa and go towards the left hypochondrial region. Because of impedance, there is a massive splenomegaly, so that will help us in differentiating between the enlargement of the spleen. So common causes of it can be either due to malaria or cirrhosis of the liver or chronic myeloid leukemias. So in various other conditions it can be called. Those were some of the common causes of massive splenomegaly. And splenectomy, that is, denotes a surgical removal of the spleen tissue is called as splenectomy. That depends again cause and condition of that particular lesion. So usually performed in ruptured spleen or accessory spleens. They undertake this procedure, surgical procedures anytime. So that is about the spleen, the topic related to the spleen. So very important, either 5 marker or can be 10 marker question, very commonly asked again. So the next topic would be the celiac trunk. So we will go to that. It's a very short topic. So that is also equally important from examination point of view. So this topic again, likewise under what headings you can learn this topic. It's quite simple, being commonly asked as a 5 marker question. So keeping that in mind again. So this is a 5 marker. So you are supposed to know the location and how it divides, like the relations of this particular. So it's a short note question. Celiac trunk as a direct question. So keeping that in mind. So we'll go through this particular topic as well. Quite a small topic. Won't take much of your time. So now this is now the introductory note where exactly the celiac trunk is located. As you can make out, this is the image of the abdominal aorta terminating into terminal branches. You can make out here. So this is how the celiac trunk artery emerging from the anterior aspect of the abdominal aorta. Now that is mostly it is a very short trunk, very short on the smaller trunk, less than 1.5 centimeters. So almost approximately 1.25 centimeter long. So very short trunk, immediately upon its origin divides into further branches. But what location wise, where on the abdominal aorta, so you have to mention it around almost about the first lumbar vertebra, or between T12 and the 12th thoracic and the first lumbar vertebra is considered to be the where the celiac trunk normally arises from the abdominal aorta. So this being a very short trunk, so immediately divides into 3 terminal, you can say 3 branches. So celiac trunk, as you can see over here, immediately after its origin, it divides into 3 branches as to be as the left gastric artery, the splenic artery, and the common hepatic artery. So these are the 3 terminal branches of, or you can say the branches of the celiac trunk, left gastric, common hepatic, and splenic artery. Now, before we look into the slight in detail about individual branches, what exactly, just for your better understanding, so what are all the, from where exactly the, this you might learn in your other classes as well. I thought just for your better understanding. Now abdominal aorta, it gives 3 sets of branches, considered to be 3 unpaired, 3 paired lateral branches, and 3 paired. Now first if you look into the 3 unpaired branches. Now 3 unpaired branches are celiac trunk starting from 1. Number 2, superior mesenteric artery and inferior mesenteric artery. These are the 3 unpaired branches, ventral branches arising from ventral aspect. So 3 paired are mainly to the 3 organs, as you can see. The suprarenal gland, paired suprarenal, a paired to the kidney, and a pair of gonadal branches. So these are three paired lateral branches. Whereas the other set, the third set of branches would be paired posterolateral. So that's what the branches that you are seeing here. So they are called as the paired posterolateral branches. Included among these three sets, you can even say the terminal branch of the abdominal aorta to be terminating into common iliac arteries. So that is a brief word so that you can get a better imagination from where the celiac trunk arises. Now we'll take up the slightly brief description about three branches of the celiac trunk. We have seen that celiac trunk divided into left gastric, splenic artery, and the common hepatic artery. First, if you take up the left gastric artery. So as you can see over here, immediately after its origin, left gastric artery almost runs up to the left and will be in close approximation. Will be running in close approximation to the lesser curvature of the stomach over here. So this the left gastric artery being in close approximation to the lesser curvature of the stomach, it also ends up giving rise to certain branches called as around the cardiac region of the cardiac end of the stomach, as to be the esophageal branches, and rest of them are the gastric branches, as you can see over here. Before the left gastric artery getting an ending by anastomosing with the right gastric artery over here. So that is how the course of the left gastric artery can be mentioned. Same image as you can make out over here. Celiac trunk arising from the left gastric, courses along the left curvature of the stomach, gives esophageal branches around the cardiac end of the stomach, then gastric branches, and ends by anastomosing with the right gastric artery. So that is how the left gastric artery can be described. Whereas the common hepatic artery, so this one, operates emergence from the celiac trunk, runs towards the right hand slightly forward and towards the right aspect of the body. So till it reaches, till it comes in close approximation to the first part of the duodenum, which is not shown over here. So imagine that the first part of the duodenum will be relation, so it moves towards the first part of the duodenum, and then when the common hepatic artery comes in close approximation to the porta hepatis of the liver. The porta hepatis is nothing but the hilum part of the liver. So when the common hepatic artery comes in close approximation to the hilum, you can imagine there will be a liver on the right hypochondria, very huge organ. So liver will be almost imagine with the liver over here. So when the hilum of the liver comes in contact, comes in close approximation towards the porta hepatis of the liver, this common hepatic artery further divides. Common hepatic artery, as you can make out over here. This region, just for a better understanding. So celiac trunk, common hepatic artery. Now this is the porta hepatis region. So wherein the hilum of the liver. So almost when it comes in close approximation to the hilum, so it ends up dividing into these three branches. Now this is like one more image. So you can make out here the lesser curvature. The fold of the peritoneum is called lesser omentum. So you can make out over here the hepatic artery, part of the hepatic artery. So that is the way it will end up dividing into three branches again. The right gastric artery, as you can see over here, common hepatic artery, dividing into right gastric artery. This is the gastroduodenal artery, number 2, and rest of it continues as a hepatic artery proper. So these are the three branches from the common hepatic artery. Common hepatic artery runs to the right. When it comes in close approximation to the porta hepatis, it will end up dividing into three branches as right gastric artery, gastroduodenal, and continues as a hepatic artery proper. So again, this if you trace the right gastric artery, it runs across again towards the left side, close approximation to the lesser curvature, gives branches to the stomach, and ends up anastomosing with the left gastric artery. Whereas if you trace the gastroduodenal artery, so this runs behind the first part of the duodenum, and ends by dividing into two branches, as you can make over here, as the right gastroepicloic artery and superior pancreatico-duodenal artery. So this will supply again the stomach, as you can see, right gastroepicloic artery supplying branches to the stomach, and again this right gastroepicloic artery will end by anastomosing with the left gastroepicloic artery, which is a branch from the splenic artery. So that is how it will end. For the superior pancreatico-duodenal, it will supply the pancreas as well as the duodenal part, duodenum of that particular region. Whereas the rest of it, if you trace the hepatic artery proper, so the third, I can say, the division of this one, common hepatic artery dividing into right gastric, gastroduodenal, and continuing has the right and left hepatic artery. And one more thing to be noted is the right hepatic artery is the one that gives rise to a branch to the gallbladder, that is the cystic artery over there. So as you can make out over there. So those are the branches arising from the common hepatic artery in short. Whereas the third artery, a few words about this one. So splenic artery is considered to be the largest among the three branches. We know that celiac artery trunk gives rise to three branches, has to be left gastric, common hepatic and splenic. Splenic is considered to be the largest, having a tortuous course. Now this splenic artery, as you trace it further, so it will end up dividing into terminal branches. So terminal branches or segmental might be two or maybe three segmental branches. Now this splenic artery, apart from being supplying to the spleen, apart from being supplying blood, primary source of blood supply to the spleen, it also gives, you can see, about the three sets of branches. Has to be pancreatic branches, the short gastric branches, and gastric fluid. So these are the additional branches. If you can, when you go through the splenic artery, see that the branches from the splenic artery. Splenic artery no doubt supplies spleen, but also has branches in the form of pancreatic branches, which are again for descriptive purpose divided into three types. Pancreatic branch can be, so this one has been labeled as a dorsal pancreatic artery. So this one, so dorsal pancreatic artery, the greater pancreatic artery or pancreatic magna, and one more has to be the caudal pancreatic artery. So these are the three sets of arteries found to be arising from the pancreatic branches of the splenic artery, whereas the second set of branches from the splenic artery is the short gastric artery, and the third would be the left gastric fluid artery. So this is anastomos with the right gastric fluid artery. So that is how the splenic artery portions of that we have seen. So that is about the splenic artery. So now this is just for your better understanding. So this is how the splenic artery division, artery branches, three sets of artery: pancreatic, short gastric, and left gastrophilioid. Among the pancreatic, these are the three subdivisions of the pancreatic branches that we can remember. So that is about the topic related to celiac gland. And last but not the least would be a rapid run through the MCQs, just for maybe for academic point of view. So now this is like number one. This question is like all the branches of celiac trunks are all except all of them are celiac trunk branches, but the exception is, so this is the exception being the right gastric artery. We know that left gastric artery. So right gastric artery is a branch from the common hepatic artery. So that's why all of them except number one. Number two, incorrect about the splenic artery. If you describe the splenic artery, we know that it is a tortuous course. That is correct. Now divides into two or three segmental branches of the high level spleen. That is also correct. Which is incorrect. Now is the smallest branch in the celiac. Now this is considered to be the largest branch in the celiac. So this is incorrect. The short gastric artery or branches of the spleen. So that is also correct. Third, which is the correct statement regarding the spleen again. Correct statement would be spleen is palpable even in normal individuals. So that is incorrect. Not palpable in normal individuals. Removal of the spleen does not impair the immune response seriously. So that is the correct statement. Even after resection of the spleen, segmental resection of the spleen, so immune response to that particular individual is not hampered to that much, to that extent. So this is the correct statement. So this says spleen develops from one mass of mesenchyma is incorrect. Splenic artery divides divides the spleen into multiple segments. It is also considered to be incorrect to some extent. Only two segments as to be in the inferior segment. So all are correct with respect to the visceral relations of the spleen. We know the tail of the pancreas, anterior surface urinary bladder or anterior surface of the left kidney. So obviously urinary bladder is not related to the spleen. So with respect to Harris rectum, base 7 ounce, is it 10 inches or in length of 5 inches? So it is incorrect. 10 inches is the incorrect statement. All are correct regarding spleen. It is an intraperitoneal line, mind you. So this is a incorrect statement. All the rest of them are correct. All these are correct. This is the one which is incorrect statement over here. So since that being a intraperitoneal, peritoneal, and the name structure marked as number 7. So what we can label this one. So this one, this is tortuous course. That is the marked as 7 is the splenic artery, and identify and label the structure that is marked as 8. So this one here. So this represents the branch from the abdominal aorta, celiac trunk. Name the visceral related to the marked area. So this is the gastric impression area, and one more last this topic. So the spleen as well as the celiac trunk. Thank you very much.
Very good afternoon to everyone. It's a vertical integration program from the Department of Physiology to the clinical side, which is integrated with surgery. And they have taught you about stomach, esophagus, and a few clinical applications will be taught here. The main topic would be endoscopy. We will be teaching you about endoscopy. Dr. Vikram, Assistant Professor in Department of Surgery. Further moving, man has evolved from ape. So is the technology and the research and development and improvement of providing an advanced medical interventions with minimal trauma to patients during surgery. Endoscopy deals with one such entity in the technology which has been applied in medicine field. What is an endoscopy? An endoscopy is a non-surgical procedure used to examine a person's digestive tract. Using an endoscope, a flexible tube with a light and a camera attached to it, the doctor can view pictures of your digestive tract on a color monitor. As the previous slide showed, that there is a tube which is inserted into your gastrointestinal tract, the upper gastrointestinal tract to be precise, and you can see the images on the computer or the TV screen. Now everything is maneuvered and controlled by the surgeon on the right side of the picture as you see here. So further moving, unlike many other medical imaging techniques, endoscopes are inserted directly into the organ. Furthermore, what is the principle of this endoscope? A fiber-optic endoscope uses a bundle of thin glass fibers. And you might have read about optic fibers, in your 11th and 12th. So this is the application of that to. I hope you can hear me now. Am I audible? Hello. Sorry. Hello. A fiber optic endoscope uses a bundle of thin glass fibers, transmits light to and from the organ to the view. Now it's just like you're walking in a dark room using a torch, and you get to see what you want when you maneuver the torch in your hand. So it is in a simple word to explain you how endoscope work. These fibers use the principle of total internal reflection to transmit almost 100% of light entering one end to the other end. Now what is the setup of an endoscope? An endoscope can consist of a rigid or a flexible tube. Now in my upcoming slides I will be showing a picture demo of how an endoscope looks. So it will be much easier for you to understand then. So it consists of either a rigid or a flexible tube. The flexible tube is one which we use these days. The rigid is now obsolete. A light delivering system to illuminate the organ or the object under inspection. The light source. The light source is normally outside the body, and the light is typically directed via an optic fiber system. Now that has been explained to you in the previous slide, where it says it works on the principle of total internal reflection, where 100% of light is traversed from the source to the object to be seen. That is your target site. A lens system transmitting the image from the object lens to the viewer typically relays lens system in case of a rigid endoscope, or a bundle of optic fibers in case of a fibroscope. Now, we are more into fibroscope these days. Fibroscope is one which is much flexible. Then you can view the image, the target image, through an eyepiece. Now this is again an obsolete part which is not used these days. This is rather seen on a TV monitor, and it is called as a videoscope, where the images are transferred to the monitor of a TV. Now with no endoscope, a camera transmits an image to a screen for image capture. So this is a videoscope. An additional channel to allow an entry of medical instruments and manipulators. Now these are the interventions or the diagnostic tools which are embedded in an endoscope, which further will be elaborated in next upcoming slides. Now how does an endoscope look? Now this is an endoscope. The entire setup is not shown. The entire setup consists of this outside where the surgeon maneuvers this instrument. This is the endoscope, a typical endoscope. And all what we see as an image is seen in the monitor here. So now coming back, this is an endoscope. Now the parts of the endoscope are well defined here and they are labeled well. I'll try to explain and make sure that you understand it in a much better way. Now an endoscope has a surgical or a surgeon's control section. This is that. Now the distal part of the surgeon control section is the one which enters the body of a patient or through the mouth of a patient. Because I am talking about gastroscope or endoscope or OGD, that is esophago-gastroduodenal scope, as it is called. All the three are synonyms. Now this surgeon's control section is where the hand of a surgeon is here. Now he maneuvers what? You have two knobs which controls this distal bending section where you can make it up, down, right and left. Now the up and down is seen with the bigger circular knob. The smaller knob takes care of the right and the left maneuvers. Now you have a suction valve, a vent hole which has an air or a water valve. Now what are these suction valve or an air or a vent valve? Now first to talk about air or a water valve is a valve when you enter into stomach. As the definition of even esophagus goes, it's a collapsible muscular tube about 25 centimeter in length. So when something is collapsed, you need to expand it to see the pathology or the normal anatomy of that particular structure. So while you are inserting the tube, you make sure that you close this valve and the air enters through the tube, through the distal vents which can be seen here, which I will be discussing later in my next slide. So as you close the vent here with your finger, the air is pushed from the scope, from the outside to the inside, and that opens up. Then you can define the anatomy of the structure which you are viewing. Later, if while going through your gastrointestinal tract, if there is any food particle which gets, which obstructs your view, then you have a water system. So if you press the same valve, the water is flushed and that particular, it might be even blood or food gets removed from the distal end, and you can view the target organ or the structure. Now suction valve, now this is a contrary to the air and the water valve. Now since you have pushed air, you are supposed to remove it, or else the patient will have discomfort, just like how a patient will have an acid peptic disease or a bloating sensation. So while you are removing the tube outside, once the procedure is done, you are supposed to suction all the air which you have inflated inside the stomach or other structure like duodenum or the esophagus. Now coming to the proximal part of the surgeon's control section, you have an universal cord or an umbilical cord which connects to the main, the junction or the setup where it is connected to the light source or the video monitoring connector. This is the video processor, it gets connected to a video processor. Now this thing gets into a video processor where the light source makes sure that the light traverses through the optical fibers with 100% total internal reflection to reach the distal end where you can see the target organ or the structure. Now, meanwhile, when you have a connection with the light source, the light traverses from here, you have a connection to the video processor where the images are transferred to the TV monitor from this rent, and the rest, the air supply and the water supply which you use here in the air valve or the suction valve, will be provided through these, which are connected externally for a water supply or an air supply. Now this is the entire setup where you can the images are seen on the TV monitor. You have a video processor, as it's been shown previously, where the videos are captured or the images are captured in the lens and they are processed here to reach the TV monitor. Meanwhile, you need a light source. Usually xenon is the one which we use right now, which is about 3500 Kelvin. Gives you an adequate illumination to watch or to see the target lesion or the structure. Then you have a suction pump, which I told you when you are retrieving the instrument or the endoscope, you are supposed to suction all the gas inside. As we move ahead, this is an imaging, more of physics. I guess this is not needed for you right now, which will be dealt with you in Department of Surgery when you enter into your clinicals. Now, endoscopic process. Now, what is this process? Now, endoscope has a channel through which a surgeon can manipulate tiny instruments. Now, I'll get back to this particular image where you can see a biopsy valve. This biopsy valve or a biopsy channel is a channel where we insert an instrument to maneuver what we need. For example, if we need a biopsy, we need to coagulate a bleeding vessel. So we insert an instrument through this rent or a channel and we can manipulate it. For this, of course, we need an assistance for a better and a convenient process. So the endoscope also has a channel through which a surgeon can manipulate tiny instruments such as forceps, surgical scissors and suction devices. As I told you, you need biopsy, you can use that rent, which further will be shown in a video where we take a biopsy of an peptic ulcer. A variety of instruments can be fitted to endoscope to different purposes. That will be elaborated as you enter into clinical site. A surgeon introduces an endoscope into the body either through a body opening such as mouth, the anus, or a small incision in the skin. The one which enters through the mouth is called as an endoscope, a general nomenclature, or it can be an esophageal gastroduodenal scope or a gastroscope if it is restricted to stomach, or one which is entering through the anus is called as a colonoscope. Now this is a diagrammatic representation where you can appreciate that the endoscope enters the mouth of the patient, enters through the oropharynx, then the nasopharynx, the distal part of the nasopharynx rather, then the oropharynx, then you enter the upper esophagus, mid, lower, then you have an OG junction, distal to which you enter the stomach, and then you search for whatever lesions is suspected, and then you target that, and that can be elaborated here in the insert, where you can see the interior of the stomach which is shown. This is the area, the field of vision, which the part is illuminated by your light source. This is a normal one. If you have a lesion where you need to take a biopsy, the biopsy channel is used and that particular part is taken for biopsy. I hope things are clear and I am not hurrying. Hope you are able to understand. Now this is the distal part of the endoscope. The distal part of the endoscope, which was not shown here in this particular slide. The distal tip. This tip, there are many rents where the lens is placed, the light source is placed, you have a biopsy channel which is placed, the opening of the biopsy channel here. This is much bigger because you need a bigger chunk of tissue for examination, and the one which is written here as B is nothing but the light. So the light is on the either side, and A is the camera or the lens where you can see the image. It's just like driving a car. You have two headlights on the either side of the windscreen from which you can drive. So it is the same process. Meanwhile, the CC is the one where you have air water nozzle. As I told you, while you are coming back, you are removing or retrieving the instrument, you will be suctioning the air from the duodenum, stomach or the esophagus. and the water part is when you compress that valve or the knob there to clear the lens and your light part. If there is any food material or blood which has got stuck, blood clot, when once you flush it, the water comes from this place, it washes this area and this area, so it will be much easier for you to control things. Now this is a small video demonstrating you about an endoscope. I hope it will be encouraging for you to become surgeons later, but it's tough to go about and it's not easy. Let's view it.
This will be next to the liver, the pancreas, and the gallbladder. Now, when we suspect ulcers, one of the first things that we would do, in addition to some blood work, is we would schedule an upper endoscopy. This is an outpatient procedure where you come to the lab and a fiber optic flexible gastroscope would be inserted through the mouth into the esophagus, the stomach, and look at the small intestine. And through the instrument, which is very flexible, we're able to take samples and biopsies, take video and pictures of the ulcer to see just what is happening. Again, you're sedated for the procedure, so it's relatively comfortable. And in this video here, what you see is a young person that has an ulcer in the stomach that deep.
Now this is the distal end.
This will be next to the big flexible gastroscope would be inserted through the mouth into the esophagus, the stomach, and look at the small intestine. And through the instrument, which is very flexible, we're able to take samples and biopsies, take video and pictures of the ulcer to see. This will be next to the liver, the pancreas, and the gallbladder flexible. We're able to take video and pictures of the ulcer to see just what is happening. Again, you're sedated for the procedure, so it's relatively comfortable. And in this video here, what you see is a young person that has an ulcer in the stomach, that deep crater right there in the stomach, right before the stomach empties into the small intestine. So this is in the antrum of the stomach, or very close to the pyloric channel. Many times you can bleed from ulcers, it can cause infections, it can perforate or break through the lining of the stomach and cause infections in the abdomen. And it can be quite painful, especially on an empty stomach. There are medications available to treat the ulcer. Many times a bacteria called Helicobacter pylori is the culprit and would be treated with a combination of acid suppression medications as well as antibiotics. And so we've come a long way in diagnosing gastric ulcers and treating them.
Now, since you have seen the video, it demonstrated you about a peptic ulcer, an ulcer near the pylorus of the stomach. I guess the video was quite good enough. Now these are the functions of an endoscope. Now what does it do? View the internal body parts using the lighting system, as I mentioned you, with the camera, and and to even record the videos such that it can be used for research, or it can be used for teaching youngsters like you who will be upcoming surgeons hopefully, and use of medical instruments to remove unwanted tumors, which has been demonstrated to you through the biopsy which has been shown. Enters through the body opening, and it usually takes about 15 to 16 minutes, which is much longer in a colonoscopy than in an OGD-scopy. Now this is a video of a normal esophagus, where I shall be demonstrating you how collapsed is the esophagus, since by definition it's a collapsible muscular tube about 25 centimeter. You can see the folds. As we enter, we reach the mid and the distal end. You can see the mucosal fold as we reach. So this is the entire length of the esophagus, which can be seen. Now this is the normal esophagus with no much pathology. Now this is a normal stomach. You can see the rugal folds as you go deep inside or as you go distally. Now this is antrum. Now the rugal folds disappear near the antrum, and that is the pyrolus which opens. This is a normal duodenum. This is a normal duodenum. Now this is pyrolus. We entered the pyrolus. We are in the D1. That is the first part of duodenum. You can see the mucosal fold now in a while. As we enter, this is duodenum. These are the circular mucosal folds of the duodenum. Furthermore, this is the exact duodenum. It will be much better viewed when you enter the clinical side, where I will be demonstrating even ampulla of Vater with bile, excreting through ampulla of Vater, and if you are lucky enough during your postings, you may end up even looking for any pathology such as peptic ulcer, duodenal ulcer. Furthermore, pancreatic tumors. Now this is the pathology which is seen in esophagus, that is GERD, gastroesophageal reflux disease. The regurgitation or chest burning chest is complaint which usually comes. Now this is GERD, where you can see the Z zone, that is squamo-columnar junction, is disrupted, and the columnar epithelium has proceeded further proximal. So that was gastroesophageal reflux disease. Now this is a peptic ulcer, an ulcer at the insula. Now how do you go about? This video will demonstrate you how this ulcer was seen, how the biopsy was taken through the biopsy channel, and furthermore it will also tell you how it was treated conservatively by use of proton pump inhibitors, which you may in general be knowing as omeprazole, pantoprazole, which are on the counter drugs these days in India. So we further go. This is the peptic ulcer which is seen in the incisura. Then furthermore we have advances in endoscopy. This is called as narrowband imaging, where we see the different histopathology type or the vascularity of the lesion, which helps in determining us what kind of a lesion it might be even prior to a biopsy report. This ulcer was given, the patient was given naproxen for 6 weeks. Now you can see the size of the lesion has reduced quite significantly. Now a biopsy is taken of the same to rule out any further cancerous changes if it might have been seen. Now this is the biopsy forceps which is inserted through the biopsy channel. The biopsy is taken. Yeah, bleeding is a complication. Bleeding does happen because you are doing something which is invasive. These are called as invasive things. Another scopy was done for the same patient after 11 months. Now you can see a complete disappearance of the ulcer. It's completely gone. Now this is the thing. It's the healed ulcer. This is the pyrolus, the pyrolic part. Now usually when you do an OGD, the length of an OGD instrument is about, it is much smaller than a colonoscopy. From the incisor to the OG junction, it is about 45 centimeters. The stomach is about 15 to 25 centimeters. There are 4 parts of duodenum. The first part is about 2.5. So is the last part. In between you have, this is another biopsy taken, the third and the fourth part of the duodenum, which usually measures about 10 centimeters each. And then you have, the large and the small intestine. The small intestine varies about 2.75 into 250 meters, and the large intestine. Yeah, now this is colonoscopy. The next video will be colonoscopy. Colonoscopy is a similar instrument like that of an OGD scopy. The instrument is much longer and in terms of length and diameter, but it works on a simple and a similar principle as that of OGD, that is total internal reflection. And the further, the talk was given previously about the principal part of an OGD. Similar or the same thing applies even into this part. Now this is a colonoscopy. This image or this video is shown on the retrograded part. They have already inserted the colonoscope from the anus to the cecum. Now they are retrieving the scope, and while retrieving you can see structures, or you can appreciate structures much better than when you are inserting. So this is a retrograded video, so don't get confused. So as the video is being played now, you will have an open through the video, and you can see it and you can appreciate it.
In the middle of the image, which is actually the opening of the appendix, and the scope is being pushed into the valve, or the ileocecal valve, and this is the opening into the small intestine. In fact, what you're looking at now are actually the villi, or finger-like projections, that absorb nutrients from the small intestine. This small intestine is known as the terminal ileum, or the last part of the ileum, which then connects into the colon. The scope is being withdrawn now from that terminal ileum, and it's going to bounce right back into the colon, which is where we're at now, looking again at that appendix opening. And the scope is now going to start looking for very flat polyps and protuberances that could potentially have the risk of turning cancer. The key here is that the colon is so well prepped that these flat polyps, or pedunculated polyps as they're called, would be seen on this visualization with the colonoscope. We're looking around each fold. You can see the beautiful vascular pattern. That's very healthy and normal. And every time we look around each fold, we're looking for something that could potentially be different from the surrounding tissue. It could look like a bump, or it could look like a change in that vascular pattern. And if we saw that, we would want to remove that and keep that from ever developing into cancer. And that's really the way colonoscopy prevents colon cancer, by looking for these polyps. The images are spectacular. This is an image of the liver, actually, where the liver is pushing on the colon. You don't actually see the liver, but you see the impression of the liver, known as the hepatic cluster. As we pull back now, we're in the transverse colon, or the middle of the colon, and you can see it has a triangular appearance. There's no cartilage or bone or anything in the colon. This is all being kept open by air that we're putting in through the colonoscope, and the triangular shape is actually just muscle that is holding it like that. It's really very floppy and very malleable, and it allows the scope to really move it about as it looks around. This is a very healthy colon, and again, you can see how clean it is. Once again, the importance of that valve prep. The cleaner the prep, the better it is to see polyps, and what we are most concerned about are flat polyps that would be difficult to see, especially if the valve prep isn't perfect. Now, as the scope is being withdrawn, there are knobs on the tip of the scope that allow the tip to be deflected up and down, and there's also a suction through a small hole that runs through the scope that we can also access the inside of the colon with different devices if we refine something like a polyp, or if we want to take biopsies. So really the patient at this point is not feeling anything at all. They're sedated, they're probably sleeping, and this whole movie, which Robert Williams called Your Own Private Discovery Channel, is really going on with them completely unaware of it. But often patients find the images quite interesting. And, the human body is really spectacular. We're at the bottom of the colon now, known as the sigmoid colon. You can see how the colon tends to close down very easily as the scope gets withdrawn. This is the way the body maintains continence. It's not a straight shot. The sigmoid colon, almost by the name of it, is the shape of an S. And that shape keeps people continent so that stuff doesn't come rushing out. And here you can see as this video is coming to a close, that we're coming towards the very bottom of the colon.
Yes, that was the entire colon, with the parts of colon being explained. You can see the mucosal folds, which are much triangle than compared to those circular in duodenum. So furthermore, yes, of course, you will be entering the clinical site. A live demo will be shown, maybe if given. If you are competent enough, I may give you a chance to do it. Well, let's see to it later. What are the limitations of the endoscope? There has been no instrument or a device which has been yet invented which can completely show us the features or the mucosal layers or the pathology in a small intestine. Either we have confined to the upper digestive tract till the second part of the duodenum, which can further be pushed to see the third part from the OGD scope. The colonoscope we can insert till the terminal ileum. What about jejunum and ileum? There has been no such device which has been invented which can be seen, but there are research and the technology is being used. One which was used recently was a capsule endoscope, where you have to take that capsule just like any other tablet, and then it goes around your small intestine to give you images. But the problem with that was you have to see only images which the capsule shows you. So there was a lot of misinterpretation and we could miss a lot of pathology of small intestine. Hence there is further research going on for the same. Hopefully we could get something which will make us much easier to view and visualize the smaller intestine, especially the jejunum and the ileum. Now compared with the other parts of the digestive tract, the small intestine is difficult for doctors to access. While a camera tip slip down the throat can get images of stomach and a tube at the other end. The end of the tract reveals the large intestine. No such devices reaches into the most of the small intestine. So the doctors rely on externally generated images. Now what do you mean by the externally generated images? These are nothing but your ultrasonograph, CT and MRI. Now what are the risks which are involved while you are doing an endoscopy? The main risk remains of infection. Yes, if you have done an endoscopy on a patient, you are supposed to sterilize it before you use it on another patient. It needs a dedicated teamwork, a dedicated sister who keeps things clean, make sure it is monitored well, and all the channels are cleared of blood, mucosa and food particle or fecal matter in colonoscopy. The most of it are done under local application of lignocaine spray, that is local anesthesia on the throat to avoid gag reflex. Some patients with comorbidities or some who are reluctant but still they have to undergo endoscopy need sedation. So there is side effects of anesthesia. A perforation, yes, cricopharynx is the smallest part while you are doing an endoscope. So that cricopharynx is a part which you have to be very careful, and that happens in the initial insertion of endoscope. And further down, stomach or esophagus can also be a part of perforation, but they still have a lower percentage of perforation if done correctly. Bleeding, yes, of course, while you are taking biopsy, if it is a cancerous lesion, they tend to have high vascularity and they start bleed, then you have to do an intervention. All the perforations are generally require surgery. Now this applies to a larger perforation in that of esophagus or cricopharynx, where there can be a sudden death of patient. But a certain minor perforation can be dealt conservatively with antibiotics and intravenous fluid. Occasionally the site of sedation or injection may become infected. This is nothing but an intravenous connection where you tend to give your anesthesia. So patient may tend to have a thrombophlebitis later, but that can be dealt with warm compression and a few analgesics, and that will settle down the pain and the inflammation at that site. What are the medical uses of an endoscope? Now I have broadly classified it as where is the indication of an endoscope. The endoscope, if a patient is having persistent nausea, vomiting or an abdominal pain which is more upper, then it's going to be an OGD scope. If it is lower, then it's going to be a colonoscope. Difficulty in swallowing, these things are seen in CA larynx, CA esophagus, CA G junction, CA stomach. Stomach is more, it presents more with nausea and vomiting than with difficulty in swallowing. Then you have gastrointestinal bleeding, any aggressive cancer, polypoidal or ulcerative type shall bleed, and they may come as hematemesis too. Now what is confirmation or diagnosis? It is diagnostic endoscopy. This is the very trivial usage of an endoscope, which includes, if the patient is having anemia and is being treated with a lot of iron, folic acid, but still is anemic, then you have to suspect that he is bleeding from somewhere. So you do both colonoscopy and endoscopy. Maybe you may end up finding any lesion which can be later treated either endoscopically, or maybe it might need an open surgical procedure. Inflammation and cancer, so you take a biopsy of any chronic inflammatory condition. Gons disease, which is an inflammatory bowel disease, might need cancer, GIST, that is gastrointestinal stromal tumor, lymphomas, squamous cell carcinomas. You have many other carcinomas, adenocarcinomas. These carcinomas are more prevalent in India, the squamous and adenocarcinoma. So, you take a biopsy and then you get an opinion from your pathologist suggestive of cancerous growth. Then you can further intervene surgically with all chemo and radiotherapy to be given. It is a multidisciplinary treatment anyway. Now, what are the therapeutic interventions which can be done in through endoscope? There are a lot of interventions which have come. There are a few benign conditions which can be treated through endoscope, like Mallory-Weiss syndrome. You have, any bleeding in stomach, like a peptic ulcer which is bleeding, or a stomach ulcer in a layman term, and you have, you can place a stent where there is an advanced growth of cancer. Though it is not therapeutic, but it can be considered under palliation or palliative therapy. Furthermore, there are much advanced procedures which are done in endoscopy now. It is called as POEMS. POEM is pre-oral endoscopic myotomy. It is done for esophageal achalasia cardia. Now all these things shall be dealt with you in depth when you come into surgery, when you in your surgical postings or in your surgical theory classes. Whatever I have been talking about to you right now is a very brief introduction of an endoscope, but very basic, and of course it will at least make sure that the window to endoscopy is open and you will never be unknown to this field of surgery. Now further to add, these are types of endoscope. You have gastroscope, you have colonoscope, you have hysteroscope. Gastroscope and colonoscope has already been discussed now. Hysteroscope helps you in seeing uterus, proctoscope, the anal canal, cystoscope, your urinary bladder, bronchoscope, your trachea and bronchus, laryngoscope, again larynx, nasopharyngoscope, upper part of the respiratory tract. Now what is the conclusion now? Whatever has been explained, demoed to you now is very superficial on a plain note. Now further things will be dealt, again I will be stressing, because it is much practical and you will be more oriented to endoscope when you enter into clinical site, and it will be demoed to you. Now as a part of conclusion, endoscope makes certain surgeries less invasive, so there is less loss of blood and the patient gets healed from his disease at an increased rate, so there is less number of hospital stay. So the patient expectations and the desires should also be considered. If he is demanding an endoscopic surgery or an endoscopic intervention, the gastroenterologist should be of that competent, and the institution or wherever you're doing should also provide you all the infrastructure to carry out with the same. The thoracic surgery to remove a locally, a tumor located near the thoracic cavity can also be removed. Now you have an hilar lymph node which can be taken for a biopsy through an endoscope, and it can be sent for lab evaluation or histopathological evaluation, and that henceforth will further encourage us to give a right and a proper management protocol. I hope this session of endoscope about endoscope will be, it is quite educative, and will make sure that further vertical integration, which is included in your curriculum, will be given a further stress of a bit of clinical exposure. If you have anything to say, any comments, you can do it. Thank you.